Okay, so it's exactly 9 a.m., so maybe I will start perfectly on time, if it's okay for you. So, first of all, thank you very much for being here. I'm Danilo Bonsenso, and sorry that I asked you to travel and come in person. I know many people are coming from several European countries, from the US, from Australia, and I really appreciate your willingness to travel that much to join this meeting. I thought it was very important for us to find a moment to meet in person and see face to face, because I see here people that really inspire me for last 4 years of research, and probably the next 30 years, and probably also people who we have all been inspiring each other. So it was very important for me to kind of having a meeting to renew the enthusiasm. We've been fighting a lot for the disease, patients to recognize themselves, but I think also us as doctors to recognize our work and our patients' diseases. So as we are fighting against even the politics, the lack of funding, the lack of recognition even from our colleagues sometimes, and this is also challenging for us to find energy and to keep going. I thought this kind of a moment would be very important to renew enthusiasm, to share ideas, and to kind of fuel energies to keep going, because the things we have to do are very important. So really, thank you very much for traveling that much. Some of you for a few days, and like flying 2 days as well. So really, I'm grateful. I can't say how much I'm happy to do this with you today. So as we are on time, we have many also patients representatives from Canada to Europe, and several doctors that have either already established long COVID or mini CFS centers, or are aiming to establish new centers, like I know in Northern Europe, or here in Italy, or even in the US. So it will be a great chance to inspire each other. We will have a very friendly and polite meeting, so feel free to talk to each other or to interact with the speakers for any doubts, and hopefully this day will be a nice day for us all. So thank you very much. I will call Paolo Palma to moderate the first session. Paolo Palma is a professor of pediatric immunology at Bambino Gesù Children's Hospital. We collaborated a lot. We will present some of our data in pediatric long COVID and other post-COVID spectrum in children. So thank you again, Paolo.
So, good morning, everyone. It's my pleasure to be here today. I would like really to thank the organizer, but overall Danilo. So I want to thank Danilo for organizing this event. Danilo actually is a great scientist, I think, because he has the ability to gather together scientists in the field, and all in Italy we recognize his ability to him, even let's say we acknowledge him for being one of the pioneers in the field of long COVID. So we, all of us, we are grateful to Danilo for really developing and moving forward this aspect of long COVID. So I want to stop here and leave the floor to Professor David Putrino. Professor David Putrino, actually from Mount Sinai Health, and I think again it's a great opportunity to listen to you. So we are really grateful for you to be here. And I don't know which is actually the speaker page. Is going to be here or okay.
Let the speaker stay up as you wish.
Okay. Okay.
Oh, perfect. Thank you. Is this advanced slide?
Yes.
It's the immunobiology.
Immunobiology.
Italian I know. Okay. Yeah, of course. Well, Daniela, thank you so much for having me here, and it's an absolute pleasure to be here. My name's David Petrina. I run a long COVID center at Mount Sinai, and we do kind of a hybrid research and clinical center. So we're always running clinical trials and trying to implement what we're seeing into clinical practice as quickly as humanly possible. That's really the mission of all of the centers that I run, including the Mount Sinai, the newest center, which is called the Cohen Center for Recovery from Complex Chronic Illness. I was really excited when Danilo invited me to be here because I've been watching his work in pediatric long COVID for so long and really been admiring it. I'll be talking a little bit about what we're seeing in adults, but with the understanding that these are studies that need to be replicated in kids because we're very clearly seeing immune sequelae that are abnormal, immune sequelae that need to be corrected, and we can likely assume that even if the immune sequelae are not the same in kids, there are immune sequelae that are occurring that need to be studied because currently they're not being studied. So with that, a lot of the work that I'm going to present here today is a collaboration between myself and my team and Akiko Iwasaki's team from Yale. We've been working together since so late 2020, and I'm really grateful to Akiko. I'm up here, someone who has no business talking about immunobiology, thanks to Akiko. And she just cold emailed me in the middle of the pandemic when she saw some of the work that I was doing and said, hey, we need to collaborate. And I almost didn't reply to her because it was a cold email. I had no idea who this person was, but something in me was just like, just Google her name. And I Googled her name and I was like, oh, I better reply to this one. We've been collaborating ever since, and it's been wonderful. So this is a study that we published in 2023. We called it the MyLC study, Mount Sinai Yale Long COVID study. And basically what we did here was we recruited a number of cohorts. So we had folks with a confirmed diagnosis of long COVID. We had people who were convalescent controls, meaning that they had survived an acute SARS-CoV-2 infection, and on screening with us, they had not displayed any problematic symptoms that potentially could indicate that they had long COVID. Interestingly, we screened a few hundred people for this who said, yep, I'm fully recovered, and we had to remove them from the trial because they said, yeah, I'm fully recovered. Oh, but I don't exercise anymore. Why don't you exercise? Oh, because when I exercise now, ever since I had COVID, I crash. I don't know what that's about. So we didn't diagnose those folks with long COVID because that's on them to chase their own healthcare. But it was an interesting data point that we had a lot of trouble finding convalescent controls. And then we had healthy controls, a very rarefied group these days, but these were folks who had never had COVID that we could confirm as best we could with blood tests looking for antibodies against nucleocapsid for SARS-CoV-2. We also just made sure that folks who were included in this cohort weren't, in any state that could affect their sort of immune function in a way that would be unpredictable for the study, because we really wanted to have a fair comparison of the three groups. In terms of people with long COVID versus people without long COVID, as you can see, they were a pretty well-matched group. The only thing I think that is worth noting here is the vast majority of people that we included into this study did not have severe acute COVID that required hospitalization. This was more, what we were really looking for was the phenotype of people who had had a less severe acute infection and gone on to develop quite debilitating symptoms that were affecting their daily life. We're just going to dive straight into results because we only have a few minutes together, and I want to maximize what we can talk about. So The first thing I want to talk about is just the fact that we're starting to see immune dysregulation in folks with long COVID. I want to point out that what we're seeing as a significant difference between the two groups is reduced CD4 T central memory cells, so your ability to remember how to fight off pathogens that you've already been exposed to, as well as an increase in exhausted T cells. So T cells that have been fighting things for a long time and are, starting to show signs that it's difficult to mount this consistent immune response. So a picture is being painted that at least a proportion of the people with long COVID that we were seeing were starting to show immune effects of a body that has been fighting something for a long time and is not maybe so good at fighting things off as we would hope. To that end, I think a really interesting interventional drug target opportunity that we're currently trying, we're currently in the process of getting ethical approval to try, is low-dose rapamycin. So in this particular, part of the immune dysregulation, what we see is an exhausted immune system. We see dysfunctional natural killer cell activity. We see T cell exhaustion. We see impaired memory T cell function. We also see reduced interferon signaling. All of this indicating an immune system that's in a little bit of trouble. Low-dose rapamycin. So if you Google rapamycin, you'll see a drug that is an immunosuppressive drug. That's at high dose. At low dose, it's actually a very interesting drug where in clinical trials in humans and in animals, it's been shown to boost natural killer cell activity, improve interferon signaling, and reduce markers of T cell exhaustion. So we're really excited to be trialing out low-dose rapamycin. So low dose for it, we don't have full approval on the protocol yet, but we're going to sort of, we're doing a dose escalation protocol. We'll probably get to patients to a maximum of about 4 milligrams per week. So that's, when I say low dose, that's really what I'm talking about. A single weekly dose of 4 milligrams. And we're slowly going to escalate. So 1 milligram to 2 milligrams to 4 milligrams. We also showed evidence of potential viral persistence. So in our long COVID cohort, we showed that folks with long COVID had significant increases in anti-SARS-CoV-2 IgG circulating in their body. And this was after accounting for vaccine doses and controlling for vaccine doses and how the vaccine doses could affect anti-SARS-CoV-2 IgG levels. So this is something that is indicative of a body that is mounting an immune response to a pathogen. Whether or not the pathogen is there, we can get to down the track. And certainly we've published a lot of work on the facts that there is at least persistent antigen and persistent spike that is clearly visible in folks with long COVID. But the body is mounting a response to a persistent pathogen. And so to that end, there are some drug targets that we can go after. And those drug targets can take the form of antivirals and monoclonals, and perhaps even combination antivirals and monoclonals for folks who are showing evidence of an immune system that is mounting a response to a persistent pathogen. There have now been a number of studies on Paxlovid. We contributed to one of those studies showing that across the board, when given to everybody, Paxlovid does not appear to effectively reduce symptoms in people with long COVID. I would say that from our perspective, that's not a huge surprise, given that Paxlovid is not an antiviral that does a good job of penetrating tissue, where we think a lot of SARS-CoV-2 reservoirs, where a lot of SARS-CoV-2 reservoirs are being found. And in addition, it's a protease inhibitor, so it stops the virus from replicating, but it doesn't necessarily eradicate the virus. So what we really think, and what we're really excited to get some funding for, is to trial monoclonal therapies, targeted monoclonal therapies for people with long COVID. The next bit of the work that I want to discuss is that we saw significant hormonal dysregulation, and we're going to go deeper into hormones soon. But one of the first findings was that folks with long COVID had reduced morning cortisol. A large portion of the cohort showed reduced morning cortisol. Now, again, as with everything, this was not everybody with long COVID, but it was many people with long COVID. And cortisol is a really interesting drug. It's a drug that promotes wakefulness. It's the thing that wakes you up in the morning. It also has significant immune sequelae. What was really interesting about this was the results were so strong that we even got an external group to try and sort of quantify it. And again, we were seeing even in our quantifying group, it was sort of captured a little bit more sporadically by other clinics, other clinics around that weren't our clinic. We were still seeing this low morning cortisol in a large percentage of the people with long COVID. Again, this is not everybody, but it's indicative of the fact that something's going on hormonally that I think is linked to the immune sequelae. And like I said, we'll talk more about hormonal stuff. But before we do, I just want to also discuss latent virus reactivation. So the other big finding from this first paper was that folks with long COVID are experiencing reactivation of latent viruses, things like Epstein-Barr virus, CMV, HHV, and this was not seen as clearly in our sort of healthy control cohorts. And so as we think about this, as we start to pull together an immunobiological picture, okay, so we've got an immune system that is fighting something off, showing signs that it's tired of fighting something off. We're seeing, memory T cells depleting and natural killer cell function depleting. And so now what do we see? We see opportunistic infections that were previously latent becoming active. So to that end, I would say that, an interventional opportunity for us is repurposing broad spectrum antivirals that have been used in the past. So we are currently recruiting and have onboarded a number of patients now for a clinical trial of 2 repurposed HIV antivirals, Maraviroc and Truvada. And we're very excited to really look into how these antivirals may influence the immune systems of folks with long COVID and address some of these latent pathogen reactivations. So in everybody who is enrolled into our trial, we're doing deep immune phenotyping first to identify who may or may not have latent pathogen reactivation, and then to see how that is affected by the drug regimen that they take over a period of 90 days. So very excited for the results of that clinical trial. And I never miss an opportunity to just thank the long COVID community for being incredible about research participation. I've been in clinical trials for, I don't know how many years, almost 20 years. And it's always a struggle to recruit, except when you're doing something in long COVID, and then you've got like 200 people on a wait list, within a few days. So let's talk about sex differences and more hormones, hormone-related work, because I think that this is something that's really important. Akiko and I sort of started to work on sex differences because, there was this narrative of there seemed to be more women than men with long COVID. And of course, if you look at 40 years of history of ME/CFS and the minimization that has occurred there, what we hear that, from these great researchers in the field, we hear that the sex differences are probably related to the increased incidence of anxiety and depression in women. That was what was offered as an explanation. I find it interesting because these folks hold professorship positions at all of these prestigious universities, and if you ask the first-year science major, hey, what's the difference between men and women, they might say, probably testosterone. And so, as we started to dig into symptom presentations, we started to see, well, there are some symptoms, not all, there's quite a bit of overlap in the symptoms, and by the way, if you can't read this, this is pre-printed online, but there are some symptoms that overlap, but then we also start to see symptoms that are more frequent in men and more frequent in women. Just for a nice change of pace, the red color is male predominant and the blue color is female predominant symptoms. And so we are seeing some predominance, and also, lo and behold, and we go into this a lot deeper in the paper, and again, I urge you to take a look. But long story short, TLDR, in women, testosterone levels will drive more female predominant symptoms, both severity of those symptoms as well as prevalence of those symptoms. And in men, estradiol levels will drive the predominance of male-related symptoms and the severity of male-related symptoms. And interestingly enough, we also did another analysis in the study that showed that if you're a man that is presenting with more female-predominant symptoms, testosterone seems to modulate your symptom levels, and if you're a woman who is presenting with more male-predominant symptoms, then estradiol seems to modulate your symptom presentation. So all of this is really interesting and shows a very clear link between sex hormone levels and both symptom presentation and severity. So opportunities, we could be doing a lot of work with hormone-related therapies. Our team is hoping to get something off the ground soon, although it's not really our expertise, so I would be hoping that someone else is out there thinking about clinical trials they can run with estrogen, progesterone, and testosterone therapies. I do know that some of our clinicians have been prescribing testosterone to women who have presented after an acute COVID infection with severe long COVID symptoms and low testosterone levels, and they have seen improvement. I also think that this is an interesting area, and shout out to Michelle because we were talking about this, an interesting area for key differences between kids and adults with long COVID, because we would expect that ratios would level out in terms of gender differences with long COVID diagnosis if some of these sex hormone differences are accurate. So finally, I want to talk a little bit about passive transfer. This is probably our newest stuff and our most interesting stuff and our most scary stuff all rolled into one. And it was done by Kayla Saar, who's an amazing postdoc working in Akiko's lab. Very briefly, the MyLC cohort that we were talking about before, the healthy controls, the convalescent controls, the people with long COVID, we took plasma from those folks. We purified IgG from that plasma, so we purified antibodies from that plasma. We injected the plasma into mice, and the mice then performed a series of functional tests, performance tests, to see what their balance was like, what their grip strength was like, what their sensitivity to pain was looking like. And lo and behold, what we started to see was balance and coordination was impaired in the mice that received long COVID antibodies in comparison to healthy control antibodies. What was really interesting was if you were a long COVID patient who reported dizziness as your constellation of symptoms, you were more likely to cause balance issues in the mouse. Similarly, as we looked at grip strength, the grip strength force test, so the animal is squeezing onto something, we saw that the mice that received the long COVID antibodies were weaker than mice who received antibodies from healthy controls. Again, interestingly, if you were reporting a symptom of tinnitus, I forget which hemisphere I'm in sometimes, but you were much more likely to cause muscle weakness in the mice than if you did. And finally, and I think this is one of our most interesting findings, is similarly, the mice were much more likely to experience increased sensitivity to pain if they were injected with IgG from people with long COVID versus healthy controls. And once again, if you were someone with long COVID who reported new onset pain, your antibodies were much more likely, almost exclusively, to cause new onset pain in the mice, even compared with people with long COVID who did not report new onset pain. So this is a really scary finding because, of course, when people get interventions like IVIG, what are we doing? We're transferring IgG from a donor into someone who is sick. And we're not really doing a whole lot to screen people who donate the plasma for IVIG infusions, beforehand. And so one of the things that Akiko and I have been talking about is the fact that when you speak to patients about, hey, did IVIG help you? Some patients say it was the best thing ever, it's really cured me. And other patients say, I really wish I'd never done that. It was the worst thing and it made my symptoms so much worse. And this could be a really interesting mechanism if we have a whole lot of silent sufferers of different immune-related conditions out there. This passive transfer mechanism could be at play. And also it indicates that we have functional autoantibodies that are causing problems. And so to that end, FcRn inhibitors, which are a newish class of drugs out there that can almost preferentially target autoantibody activity, could be a really great target for folks who are showing signs of autoimmunity as a driver of their long COVID symptoms. So I wanted to finish, but we had some conversations last night that spurred me to just add in a couple more slides. So sorry, I'm going to go a little bit over. Because, when we start thinking about, well, how prevalent is long COVID and, who has this, I think it's the wrong question. I actually think the right question is, we have a virus that is seemingly affecting every aspect of our physiology. I've just talked about immune stuff, but we're going to have some amazing speakers talking about everything else. And what are we doing to prevent this virus from infecting people over and over and over again? And I just want to share some work that we published in Cell a couple of years ago that was led by Michelle Mongi at Stanford. In this study, basically what we did was we infected mice with a very local version of SARS-CoV-2, so it never left their lungs. And we confirmed that the virus itself never left their lungs. But what we did see was that even though the virus never left their lungs and they were only mildly ill, it created a cascade of chemokine and cytokine activity that led to hyperglial activation, glial hyperactivation and cell death in the brain as a result of their illness that was localized to the lungs. We also saw that the chemokines that did the most damage to the central nervous system were highly represented in long COVID patients from our cohort, human long COVID patients from our cohort, who reported the most severe brain symptoms. So this study was interesting because it showed that even a mild SARS-CoV-2 infection that might feel inconsequential was doing damage to the central nervous system. This is further backed up by a study that was published in Nature in 2022, where BrainBank basically a UK brain bank was looking at brain differences in folks who had survived an acute SARS-CoV-2 infection. Crucially, patients who did not have long COVID. These were people who said, yep, I've fully recovered. And when they compared their pre-COVID MRI to their post-COVID MRI with on average, the sort of time difference between those MRIs was about 2 years. They showed cortical thinning, so brain matter loss. They showed functional network changes in their brain. They showed poorer performance on trail-making tasks, Trails A, Trails B, cognitive tasks. Overall brain degeneration that outpaced controls, healthy controls who just had not experienced the SARS-CoV-2 infection and had a MRI before and after. So what we see here, as we start to consider what is long COVID, and what should we be doing in terms of a policy, we're seeing degenerative changes that are accelerating. Now, you can make all the arguments that you want. Well, other viruses do this as well, other infections do this as well. Yes, but we're not getting, I agree. I think infection prevention should be something that we implement across the board. Unfortunately, there isn't a virus out there that is infecting people as quickly or as efficiently or as effectively as SARS-CoV-2 right now. So we need a lot of protections in place to make sure that our cognitive futures are intact, especially in our children who are not getting a lot of protections at the moment. I'll spare you the other study that showed that every SARS-CoV-2 infection comes with approximately a 3 IQ point drop, because that might be too scary, but I'll just end here by thanking Dilo for letting me go over, thanking you all for listening. These are the wonderful people that I have to thank. So, thank you.
It's my great pleasure to introduce Risa Bertolis, who is Distinguished Professor and Chair of Physiological Science at Stellenbosch University and an Honorary Professor at Liverpool University as well. And thank you for being here. You will discuss about the etolopathy of long COVID. Thank you.
Thank you very much for the introduction. Thank you very much, Danilo, for the wonderful opportunity to come and speak to you all. And it's wonderful for me to meet old friends and make new ones. And I have really enjoyed my presentation today. So I just need to thank a few of my collaborators, like Cal, and then some of my students, some of the new collaborators that I have here, but mostly my students, and one of them sitting in the audience as well. Thank you. And then also thank you for the funders that I also just like to recognize. So over the past years, our lab and our collaborators have been studying platelets as well as the coagulation system in numerous diseases. And in 2020, we turned our attention to acute COVID. And we looked at how in acute COVID at that stage, and then we progressed into long COVID, we looked at how the receptors on the endothelial layers interact with platelets, and that's how we come to the Danilo's work. And then also be able to form these microclotting when we have the presence of inflammatory molecules, and at that stage we were also focusing on the spike protein. So our work then progressed into long COVID as well, but we started off actually quite a few years ago by looking at how the clotting protein, fibrinogen, which is a soluble protein, might interact with inflammatory molecules and also the spike protein. And we have done quite a lot of work then to look at how the soluble protein fibrinogen, in the presence of inflammatory molecules, when we have direct protein interactions, can change and become an insoluble little structure or protein when it interacts with these inflammatory molecules. And we discovered probably around about 2014 until 2016 that these type of inflammatory molecules can cause the fibrinogen molecule to become amyloid. So it's not the same as the amyloid in the brain. It's not an amyloid protein as in the brain, and that we know amyloid in Alzheimer's, but it's the same effect of any protein interacting with the inflammatory molecule like LPS or other inflammatory molecules, also estrogen, and make it spike, it can change into a structure that becomes amyloid and then an insoluble little structure of the fibrinogen molecule. So when we turn our attention to long COVID quite early on in the pandemic, we discovered that all of these work that have been done by various groups, Akiku and David, if we look at what we see as long COVID with immune dysregulation, with various hormonal changes, with viral persistence, with cytokine dysregulation, we thought that we could all take it back to the fact that all of these might have effect on the endothelial layers or the vasculature. And if we look at the vasculature and all the changes that we saw early on with acute COVID regarding platelets and the changes to the fibrinogen molecule, we thought that perhaps thrombotic endotheliitis could be a key pathology of long COVID together with clotting dysregulation. Now, what do we need to do for long COVID research with all of those laboratory and research knowledge that we have? We think this is obviously a very oversimplified diagram. We think if we look at the long COVID population, we need symptom classification, but then we need to follow it up with biomarker identification to identify the subgroups or the phenotypes in long COVID so that we can eventually customize interventions based on biomarkers. So our research focus is therefore to look at these biomarker identification with various techniques, but then also perhaps to discover new ones and new different fractions of blood perhaps that we could study to use as a biomarker profile for subsets of long COVID patients. Now, just to circle back to what we found initially in acute COVID, which is obviously we're very relevant now for long COVID as well. Initially, in the first weeks, months of the pandemic, we studied platelet hyperactivation using scanning and microscopy. And here you can see platelets interacting with red blood cells, and this is a close-up of the same micrograph here, where the platelets interact with the red blood cell membrane. So severe platelet hyperactivation, seems the platelets even burst open during acute infection. And then we also noted these structures, clotting pathology, microclots. And that's like we weren't calling it microclots yet, but even gluing together the red blood cells in the acute stage. Then we looked at our fluorescent microscopy with our methods, flame T, which marks amyloid protein. And we found this example of healthy plasma where you find little microclot events, as you would expect, because no one is perfectly healthy. Type 2 diabetes, we found a little bit more. But we were absolutely mind-boggled to see the acute plasma with huge changes of the plasma becoming and forming into an amyloid shape. Now, if we just look at this microclots here in long COVID patients, and this is done, we probably look at thousands of patients now. This is still after the infection, it's now long COVID, perhaps months after. We still see these microclot structures that interact with the red blood cells. And here you can see one of these microclots in the Z stack. Just a huge structure that are circulating in circulation of patients with long COVID. And then obviously we don't want to forget the platelets, and I'm not going to spend my time today on platelets, but platelets in long COVID also significantly hyperactivated still months after the infection. So the big question that we want to address is whether microclots and their content might have a possibility or place to identify long COVID patients and even perhaps any serious post-viral diseases, and can it be used as a measurable biomarker in the clinic? So just circling back to endothelial dysfunction, we published a paper recently where we discussed what can clinicians do to look at and find endothelial dysfunction, perhaps not necessarily microclots and platelet hyperactivation, but what can be done? There can be lots done, and these are just some of the methods. It can be radiology, nail fold fluoroscopy, that can be used by a clinic to try to understand if there are endothelial dysfunction of patients. So the narrative of microclots, are they indeed clots? So I'm getting this question by some people. Why would it matter if we have got amyloid particles in circulation? Is it something that we need to look at? Does it matter that if it's present in healthy individuals, diabetes and other diseases? And our simple question or answer to this question is, it's not a reason to ignore it. And the important thing is, we need to understand what are the differences between the different diseases. What are the differences in the microclots? Is it simply numbers and sizes? Or are it perhaps something much more complex? Is it perhaps something like the content, the molecular content of microclots? So is it coming, why do we say we can call it clot? This is the definition of a true clot or a thrombus, if someone suffered from a stroke or a heart attack. And the important consideration is that you need to have fibrin in it, you need to have red blood cells associated with it, neutrophil traps, you need to have that, and you have to have white blood cells associated. This is the general definition of a composition of a true clot. So I hope I will be able to convince you that these microclots that we have been studying actually do conform to this definition. So let's start with red blood cells. This is a healthy red blood cell. These are red blood cells from patients with long COVID. The microclots are perhaps smaller than the big clots, but are they associated with red blood cells? Definitely. We have seen that they come together, they actually glue together the red blood cells. And perhaps microclots are the start of macroclots. We also, obviously we haven't spent too much time on it yet, but this is the plan soon. We spin out our microclots in plasma. And here's just an example on how the red blood cell will look like if we just take whole blood and we take the whole blood and just put THT with it, theophylline T with it. You will see microclots associated with the red blood cells. Now, early on in 2021, we looked at the content of the microclots. We did a proteomic study, and we looked at acute COVID, long COVID, and diabetes, and controls. We double digested the plasma because we couldn't digest the COVID and the non-COVID plasma with a single digestion step. We determined that the content of the microclots were quite interesting. We saw von Willebrand factors significantly increase in acute COVID and long COVID. We saw fibrinogen alpha chain. We saw alpha 2 antiplasmin. We saw beta chain and fibrinogen, all of the fibrinogen chains were therefore increased and changed, according to us, inside the microclots. What was interesting, inside the digested microclots, we found alpha 2 antiplasmin, and that is a molecule that prevents clot breakdown. So now we had a reason why the clots might be lingering longer in patients than they were supposed to. Now the question of can spike protein turn fibrinogen amyloid? Here's just some of our results. We took purified fibrinogen from Sigma. We exposed it, in this case, to spike protein. We then exposed it to alpha chain fibrinogen antibody and theophylline T. Here you can see the orange of the alpha chain where the microclot indeed changed, or the fibrinogen indeed changed into an insoluble clot. Some of the areas became amyloid, and these are overlaid in this picture. So the inside of the little insoluble microclot became amyloid. The same you could do when we added LPS to fibrinogen, and here you can see the overlay as well. So the purified fibrinogen changed into an insoluble little clot, and you can see certain areas of it can become amyloid in the presence of LPS. So we were very, very excited to see some of our collaborator work. Radio et al. Just published a recent publication where they confirmed that fibrinogen turns into a pathological structure, and they proposed that monoclonals might be able to prevent this. A wonderful publication. We also did a collaboration with Professor Alain Theory. He's from INSERM, and he looked at his interest as NETs. We looked at NPO, neutrophil elastase, circulating DNA, and microclotting, and we could look at the differences, and we could find interesting differences between the various molecules. We also found that NETs and microclots are structurally associated, and NETs might therefore be another measurable biomarker in collaboration and structurally associated with microclots. And then you can see the microclot with NPO muloperoxidase DNA parts, and then you can see the flavin T and the combination of the net and of the DNA from the net and the amyloid comes together. So we do believe that this might be an interesting avenue to pursue if we look at how the little clots look like, flavin T fibrinogen antibody inside the clot, as well as NPO, which are the blue areas here. So we do believe that this might be another measurable biomarker that we could use. And this is just another example of some of our experiments where we found NPO fibrinogen antibody associated with the microclots, confirming that the microclots from long COVID are indeed amyloid, and they do contain fibrinogen, and they are associated with muloperoxidase and NETs. So another important question that we would want to ask is biofilm. Might biofilm be a possibility associated with our microclots? Because we are in search of new avenues. We want to find new associations within these dense deposits that we call microclots. And for that we used biofilm, a biofilm marker, and we did a self-titration series. The reason why we did that is we would want to have the spectral view as distinct differences so that we can indeed combine our various markers and not see overspill between our markers. So this is an example of biofilm that we indeed found inside our long COVID samples. We see that biofilm is associated with our amyloid microclots, and this is just again with biofilm, flavin T, and our fibrinogen chain antibody. What does this mean? Does it mean that biofilm is associated in every single microclot? It doesn't even mean that it's associated. It's present in every single long COVID patient. But remember, in the beginning of the presentation, I said we are looking for phenotypes. So perhaps some individuals might have biofilm interacting and playing a role. That is what we need to find out. Which biomarkers can be used for which phenotypes? We did the same with our fluorescent microscope, as well as here our flow cytometer, our imaging flow cytometer. So we set up our flow cytometer to identify microclots with biofilm in our lab. Then endothelial damage, we are interested in endothelial damage. In this case, syndican-1 is an important marker that we believe might be of interest. We also optimize that, and we optimize it together. We can now use flavin T, fibrinogen, and syndican to detect which microclots might have osmolality associated. And again, syndican we did also with this titration series, and in our imaging flow cytometer, we could indeed find differences and very nice separation of the signal. We are obviously very, very happy that some of our collaborators have been doing a lot of other work that is associated with microclots. This is just a wonderful example of the WISP groups that found microclots inside muscle using our methods. And then Lyle, that will be talking soon as well, have been finding microclotting in pediatric patients. And then I'm very excited to share our first results with the heart of the test. With Naidu and student Liz, we have been looking at microclotting in children, and we have been using fibrinogen antibody, theophylline T and Hoof stain, and you can see the overlay of where they are. And in children, we also look at lactobacillus net coming out of the microclots. So in children, microclots are definitely present, and we have found nets and amyloid changes in the plasma of these individuals. This is just a bigger version so that you can also just see the net associated with the microclotting and the overlay. So this is the fibrinogen part. Some of the fibrinogen is called amyloid, and this is the net coming out of it. So there are other exciting research that happened, not in long COVID sphere, but that we are excited about because it confirms the presence and utility of microclots. And this was work from the TOE group that found microclots in sepsis, using our methods and our proteomics as well. So the question is, can microclots in their content be used as one of the diagnostic markers, not the diagnostic markers, but one of the diagnostic markers of a subset of phenotype? And we believe that microclot testing might be a new diagnostic frontier that we could use. Microscopy, probably not the way to go. Imaging flow cytometry, definitely, and flow cytometry, definitely. We published a paper last year where we developed the method to use microclots using imaging flow cytometer and using platelet-poor plasma. And what can we learn from this, from specifically the imaging flow cytometer? We can learn that there's a difference between the different groups, but it's not just the numbers that count. We need to split up the different size ranges. So there are different sizes in microclots. Which ones might be the most pathologically sensible to use as a measure? And probably the bigger ones, size ranges. And here we compared long COVID with ME/CRS and controls. So definitely lots of work to be done and not finished yet. I was very excited to see Peluso's work, Peluso and Diggs, where they talked in one of their Cell papers a month ago, where they talked that microvascular dysfunction and tissue hypoxia are central pathologies in long COVID. They also mentioned in this slide microclots and macroclots. And very interestingly, we now, I think, found an interesting phenomenon where microclots might be involved in the formation of macroclots in stroke. So this is very important for us to pursue further because we believe that some long COVID patients might have a bigger propensity to develop heart attacks and stroke. And this is some of our exciting work that we've just pre-printed. This is from extracted stroke. It's not long COVID stroke, it's post-COVID, but it's extracted stroke clots, where we looked at the stroke clots, sectioned it, and looked at the theophylline T staining, and we did find inside the clots significant amyloid protein changes of the fibrinogen, which suggests to us that microclots, if left untreated and undiagnosed, might develop into stroke sometime in the future. So the take-home message. The content and the activity and the biochemical characteristics are crucial. Is a microclot or a microclot? In a control, a disease patient, a long COVID, is it the same? No, it's not. The content, molecular footprint, is of significant importance. We cannot ignore the role of hyperactivated platelets. They go hand in hand and they both drive vascular dysfunction. Ultimately, these two pathologies, microclotting as well as platelet hyperactivation, we believe are the two key pathologies in dry thrombotic endotheliitis in long COVID. And we need to diagnose in order for us to determine which phenotypes are present, and only then can we progress into clinical trials when we have diagnosis to follow up if the clinical trial outcomes have been successful. Thank you very much.
It's really my great pleasure to introduce one of my friends, and also actually I think one of the greatest scientists at the moment in pediatric immunology. So Pedro Brodin is professor at Karolinska Institute, but also he has a double appointment also at Imperial College, and he will talk about long COVID and evidence from SARS-CoV to persistence. So thank you, Peter, for being here.
Thank you, and thanks everyone for coming. Especially Danilo, thanks for organizing this. You've been such a force of, for good during the whole last few years for children, and you've inspired many of my clinical colleagues to really care about these patients and look more carefully. I'm going to tell you a little bit about how I ended up studying patients with persistent symptoms, and this was actually together with Paula. Paula, we during the pandemic, or after the initial phase. As pediatricians, we started seeing these children coming into our hospitals with hyperinflammation. And they presented a little bit like Kawasaki disease, but they were slightly different age, some more severe than what we used to see. And so we started characterizing these patients. What is this? It came, if you go to the next one, it came 1 to 2 months after the initial infection in these individuals. And they had a multitude of symptoms, but often involving the heart. Many of them had shock. We had a few deaths, sadly. And we really tried to understand what this is. And if you go to the next one, we were fortunate to have an ongoing collaboration even before the pandemic, together with Paolo, looking at Kawasaki disease, which is the one condition that most resembles what these patients presented with. And so we were wondering, is this just Kawasaki disease triggered by SARS-CoV-2, or is it a completely different pathology? We used, what we often do in my lab, is to try to profile the immune system very broadly, taking all the cells and proteins into account. And if you go to the previous one. And so what we basically were able to show in this publication was that no, this is probably not classic Kawasaki disease. It is a different condition. It's triggered by SARS-CoV-2. And it involves a multitude of hyperinflammatory cell responses. And if you go to the next one, and this is how I was really inspired by the work by Dale Donker and colleagues in Boston, showing persistent virus in the gut, or leaking through from the gut into circulation. And this really sort of sparked at least my entry into the idea of studying persistent symptoms, as is there some commonality between these children that presents 1 to 2 months after SARS-CoV-2 infection, in their case with a hyperinflammatory, life-threatening condition, and these other individuals that started to appear which were having milder symptoms but persisting for a long, long time after SARS-CoV-2 infection. Could we learn something between these two groups? That was my sort of way into long COVID. And so if we think about a normal immune response to a virus, I know to many of you this is kind of obvious textbook knowledge, but it's important to keep this in mind because this is the reference by which we have to look at, long COVID immune responses. So you have an infection, you mount an initial response, which is this innate response, in this case mostly in the airways, triggering a T cell and B cell response, the adaptive immune system, right? And those are the memory responses that protect you in future infections. Now, what typically happens if you look at the purple line on the right is that there's a virus that increases quickly and then disappears, as far as we know. There's an adaptive immune response, and the adaptive immune response takes a couple of weeks to get started, but once it's off, it lasts. The B cell response, which is in blue, typically wanes over time, right? And I think all of you remember the discussions after in the initial wave of the infections. How long does the antibody response last? How long am I going to be positive? And when am I at risk for another infection, etc.? But anyway, the antibody response wanes, okay? Now, if you go to the next slide, what we did was we teamed up with a beautiful group of clinicians in Stockholm who had initially started profiling patients that were discharged from ICUs after having severe acute COVID. And they wanted to follow these individuals over time. Then that group, which was a multidisciplinary group of physiotherapists, infectious disease physicians, pulmonologists, etc., they started seeing these patients with persistent symptoms even after mild acute COVID, okay? And they started profiling these groups, and they were telling me, there's something weird about these patients because their antibody levels just keep going up and up and up and up. So what we did was we used a Simoa assay, which is an antibody assay, or looking for antibodies using a very sensitive type of ELISA. And what you see in purple and blue is up to 1,000 days after their initial SARS-CoV-2 infection. This is similar to what David just showed from their cohorts. The antibody levels to SARS-CoV-2 are so high, right? And they just keep increasing. While in yellow and orange here, you have convalescent individuals. So these individuals had SARS-CoV-2 around the same time, but then they're convalescent. So this goes against the textbook knowledge of antibody responses waning over time, right? And it suggests that something is driving this antibody response to continue expanding. Next slide. So we decided, okay, how are we going to what's our best chance of understanding the underlying mechanisms of this disease? So we've been working throughout the pandemic within this incredible international network called COVID Human Genetic Effort, where scientists across the world, 150 centers, learn from each other, share samples, share data, to try to understand different manifestations of disease after SARS-CoV-2. We decided to start a subgroup on long COVID, which I'm leading, where we try to identify the most severe cases we could find in each center. And by severe, I mean the individuals that had objective manifestations of disease, organ pathology, organ dysfunction or damage, but did not have a severe acute COVID that would explain that damage to their tissues, right? And as you know from many cohorts now, many descriptions, these symptoms vary enormously. It could be all kinds of different things, but they could be grouped into one that is immune dysregulation, one that is autoimmunity, as David showed, and viral persistence could be one of these mechanisms as well. These are not mutually exclusive. Viral persistence will likely lead to autoimmunity because chronic stimulation breaks tolerance. Go to the next one. And so here's Judith, who leads this clinical cohort, and basically what they've done is they've screened over 2,500 individuals now, and this number of individuals that have objective measures of disease, I mean, that depends on the sensitivity of your measurements, right? I'm not saying that the bigger group that do not have objective measures of disease, I'm not saying that they're making this up. They probably have some underlying pathology that we just can't detect with current tools, right? And so we had a very strict case definition, 12 weeks, objectively measured organ damage dysfunction, and we have to have a PCR verified SARS-CoV-2 infection prior to this, and we do not want to have severe acute COVID that would explain tissue damage and disease. So what are the symptoms? So this is a pretty complex heat map, but basically black means that the symptoms detected, patients are on the x-axis, symptoms on the y. Just to give you a few examples, POTS, posterior orthostatic tachycardia syndrome, easily objectifiable symptom. Cardiac symptoms, that's a mix of things, inappropriate sinus tachycardia and other autonomic nervous dysfunction symptoms can easily be measured. So there are a couple of take-home messages from this. The symptoms vary broadly, okay? There's no obvious groups. It's not like some long COVID patients, these are all adults. Long COVID patients have one group of symptoms and another group have another group. You know, you understand what I mean? It's very broadly variable. These are, just one thing to point out, these are 107 of the first patients we profiled. 87% of these were females. So coming back to that in a little bit. Go on. And you can look at the next one right away. Now, no, the previous one. So the thing about measuring in the blood antibody levels indicated to us that probably the virus persists, because otherwise these antibody responses would not be growing and growing and growing. And then other people, so this is a study from Michel Nussensweig's lab in New York showing that if you look on top there, those pie charts, those show individual B cell responses in individual patients, either at 1 month or 6 months. And what you can see is the red part of the pie is an expanded clone of B cells, so they are responding to something. Now, what they see is that yes, those B cell numbers decrease with time, but if you look at the specific sequences of the B cell receptor, the antibody, there's continual adaptation of that antibody sequence. And that will never happen if the antigen is not there. Okay? And then when they look in intestines 4 or 5 months out from the initial infection, they could find the antigen. And this was rather early in 2021. So since then, obviously, many have reported persistent antigens in various biopsy materials, necropsies, etc. Go to the next one. And here, in keeping with what Leif Jonker and team showed in MIS-C, here's a girl with persistent intestinal pain 3 months after mild COVID who did a biopsy of her intestine. And again, viruses is present. And this is not someone who has severe MIS-C or any other long symptoms, but just a mild intestinal pain, enough to do a biopsy, and the virus is detectable. Okay, I was asked to talk a little bit about how do we measure viral persistence, and this is a really tricky thing because we cannot biopsy every tissue in individuals. We would love to have a marker or a measurement in blood, and this is the one that was used in the MIS-C paper I just mentioned. The Simoa assay, I would argue, is probably the most sensitive protein detection method. It's basically an ELISA, but it's concentrating your sample using beads in microwells, and then thereby getting increased resolution. In the first representation by Schwank from the Walt lab at Harvard, there is a, you can see the bead panel there showing total spike detected in patients with long COVID as opposed to convalescent. But it's not everyone, right? So that's a really important to keep in mind. You do detect this in some individuals, not in everyone. Go to the next one. This is our cohort, and we measured using the same method, the Simoa assay. You can see the total spike, S1 fraction of the spike, or the M. And as you see, a few of these individuals are positive. This is days after infection, so in yellow are the convalescent, and in purple are the long COVID patients from 2 different cohorts. But it's a fraction that's positive, And there's also very little overlap between the different detection modalities, so S1 and total spike and M. It's not like if you're positive, you have all of them, okay? Next. We also tested an alternative assay. This is a spin-out from the Wyss Institute, I think, in Boston. And basically this company has a different type of method, which is also an immunoassay using antibodies to detect proteins of interest, but the readout is different from the Simoa one. Here it's based on real-time PCR, amplifying a product from 2 antibodies that bind S1 in this case. When we tested this assay in our cohort on the left, there's a lot of positive hits. But those positive hits do not overlap with the other assay, and they're not only restricted to patients with long COVID. So in our case, this assay is a little bit shaky still. I'm sure they're working on it. But next. And this is the latest paper from the Harvard group showing in multiple cohorts the frequency of antigen positivity in plasma samples from 4 different cohorts. I would guess this, to me, this is sort of state of the art when it comes to measuring antigens in plasma. And as you can see from the summaries on top, the mean values are 14, 15, 20% positive individuals in these cohorts. So what does that tell us? This is not because the assay is bad, but this is, in my opinion, because plasma is not the right place to look. You go to the next one. And then our collaborators in Belgium have developed this assay based on nucleotide detection in blood using a nanostream platform. And what they're showing basically is in the volcano plot in the middle on the top, there are some red dots there. Those are highly abundant in long COVID patients, and those are RNA from SARS-CoV-2. So you're detecting SARS-CoV-2 RNA, even antisense, which you would only find if you have ongoing replication. So this is another assay that's really good, but again, we're only detecting positive virus or viral RNA in a fraction of patients. So it's not going to be a diagnostic test still, in my opinion. Next. We tested this again in our cohort, so we tested all of these assays now in our cohorts. We find a few positive, very little overlap between RNA detection and protein detection, and many negative patients. Okay. So again, I think from my conclusion in all this, we don't yet have, and I'm happy to discuss this later on, in my opinion, we don't have a plasma assay that is a diagnostic test, right? That's uniformly sensitive for detecting all patients. And like I said, I don't think it's because the assays are bad. I think because some patients carry virus deep in tissues and it doesn't leak into plasma, right? And so therefore, this is probably not going to be the way to diagnose the patient. Yeah. Now, with all that said, in my opinion, having really elevated antibody levels, controlling, like David said, for vaccine exposure and so on, or looking at M antigen, which is not in the vaccine, is probably, a sensitive enough measure of viral persistence. It's an indirect measure, but it tells you something about some things driving B-cell responses, right? And so if we use that in our cohort and look at just the antibody levels and what correlates with really high antibody levels as an indirect measure of viral persistence, right? You can go on. We've looked at T-cell responses. David talked about exhausted T-cell responses. And anytime you have a chronic stimulation, T-cell responses that respond to that antigen will become exhausted over time. We see that in tumors, we see that in many viral infections. So that is probably true. Now, is that the cause of the viral persistence or not? We don't know, I would say. In our code, what we did was we looked at all the CD8 T cells. The single cells were clonally sequenced, so we sequenced the T cell receptor of all these individual T cells. And then we mapped those T cell receptors, those are the gray bubbles there. All the gray ones have specificities that we don't know. Could be a bacteria, other viruses, self-proteins. But there are a few that cluster towards SARS-CoV-2. They're SARS-CoV-2 specific T cells. Some cluster to CMV, flu, EBV, other viruses, right? Now, what about the fraction of these SARS-CoV-2 specific cells in patients with long COVID as opposed to healthy or convalescent? To keep going. If we look on the top here, we see the relationship between total clonal expansion, so expansion of T cells responding to anything, whatever it might be, in relation to the spike level of antibody. No relationship. There's no correlation, as expected, right? These two are two different things. But if you look at the correlation between having high levels of antibodies to spike and having a fraction of all your T cells specific to SARS-CoV-2 expanding or not, there's an inverse correlation. So, for those of you who are not immunologists, let's go through what this means. So you have high levels of spike antibodies. I've already told you that that's an indirect measure of viral persistence. It has caveats, but let's say that's what it is. Now, the higher your antibody level, the lower your expanded SARS-CoV-2 specific CD8 T cell is. Suggesting that maybe the reason for the viral persistence and the high antibody levels is a failure of your SARS-CoV-2 specific CD8 T cells to expand and clear the virus. That's what our data suggests. Next. We looked for exhaustion, using transcriptional analysis of these single CD8 T cells. In our hands, when we just compare patients that have high anti-spike IgG or low anti-spike IgG, we see no difference in the level of exhaustion markers. To really tell you if they're exhausted or not, we need to stimulate them and see if they respond. We haven't done that. Now, this suggests that, okay, in our case, in our cohort, we don't really see exhaustion, but we do see failure or lack of clonal expansion. Let me go through the next one. So this brings us to a hypothesis. That is that potentially, if you have an infection with SARS-CoV-2, you mount an interferon response. Let's say that interferon response is moderate. It's good enough to limit viral replication. You get CD8 T cell expansion, efficiently clearing the virus. You become convalescent. Now, let's say if your type 1 interferon response is really strong, we think of that as a good thing because that limits severity of your acute COVID. But what if that really strong type 1 interferon response inhibits your CD8 T cells to expand? Maybe you're more likely to have long COVID and viral persistence because you failed to clear the virus. You see how these things could relate. This is a hypothesis. I'm going to tell you some of our recent data that I think points in this direction. Next. So if you look at just public data from COVID-infected individuals on the top there, what you're looking at is transcriptional responses associated with high type 1 interferon response in CD8 T cells. So if a CD8 T cells exists in an environment with massive type 1 interferons, they will upregulate these genes. And what you see on the top there is that there's an inverse correlation between CD8 proliferating and having seen a lot of type 1 interferon. So supporting this idea that in the context of a high type 1 interferon response, CD8s will not proliferate as well. In NK cells, we don't see that at all. There's no such relationship. This has also been shown now in cancer patients, for example, that type 1 interferon during cancer prevents anti-cancer responses, and people are, playing around with treatments against that. Next. We also set up an experiment where we took tonsils from patients who operate out their tonsils, tonsillectomies, and then those can be cultured, and you can vaccinate the tonsil in vitro in a dish as a way to simulate an adaptive immune response, which you cannot do in the blood, okay? When we did that, in this case we had to use flu. We're trying to do this with SARS-CoV-2 now. But basically, if you do that and then you throw in interferon as an additional condition, you basically find that in 4 different donors, adding interferon to the flu virus alone reduces the amount of effector memory or expanded CD8 T cells. Again, supporting this notion that high type 1 interferon responses might actually have a negative impact on the CD8 T cells that you want to expand. Next. Okay, so this brings me into my other part of my talk, which has to do with why do some people have a high type 1 interferon response and others not. And this brings us to sex differences again. I mentioned that we had 87% females in our cohort, predominantly females of reproductive age. Other cohorts have different ratios, but it's generally long COVID is, there's an overrepresentation among women. There are other differences between male and female immune systems that we also need to keep in mind. In general, infection disease severity, and now I'm talking about the acute infection, sepsis, flu, acute SARS-CoV-2 infection, more severe in males than females. Vaccine responses tend to be stronger in females, but side effects to vaccines are also more frequent in women than men. Autoimmune diseases, often much more in women than men, 4 times more likely in females. Some diseases like lupus have 9-to-1 female to male. Go on to the next one, please. So in the SARS-CoV-2, I've already mentioned, and you all know, case fatality rates prior to vaccines, always higher in males than females in the beginning, right, when everyone was naive. We know why that is. We know that if you have a very strong type 1 interferon response in the beginning of your initial SARS-CoV-2 infection, you're going to limit viral replication and you're going to have mild disease. Right? We know that from numerous different observations. If you fail to mount a strong type 1 interferon response, you're going to be more likely to have severe disease, which is driven by a pro-inflammatory response, TNF, IL-6, a cytokine storm. Remember in the beginning of the pandemic? Which we can limit with steroid therapy. And this is due to the immune system trying to keep up with the virus that has been given a head start. Okay? Next one. The problem, we know that all of these things I've told you, these are known things, but what we don't know is if you look at males and females, and you compare male immune systems from female immune systems, which aspects of those differences are caused by hormones and which ones are genetics? Because the XY chromosome and the sex hormones, we can't distinguish those if we just look at males and females, right? So what we did way before the pandemic, since 2016, my team members and I set up a cohort where we try to follow what happens to someone's immune system when they undergo gender-affirming hormone therapy. So in this case, the genetics does not change, they stay the same, but the hormone levels go from one to the other. And by looking at what happens to an individual's immune system when your hormone profile completely switches, you can understand which parts of your immune system is regulated by hormones as opposed to genes. And so we did this, and this is the first cohort which is published, and this is 23 trans men, so assigned female sex at birth, and then receiving testosterone therapy, and then during that period we profile their immune system. What happens is that testosterone increases, obviously, because that's what these individuals are given, but at the same time, estradiol and progesterone decreases, and they stop their menstrual cycle within a couple of weeks, variably, but around that time. Now, what happens to their immune system? If you go to the next one. This is just looking at bulk mRNA transcripts. And there are basically two types of processes that change when these individuals get testosterone. The first thing is that little pink cloud on the right, left, your side, is type 1 interferon responses. These antiviral responses I just mentioned. The responses that also drive lupus, Sjögren's disease with 9-to-1 female to male preponderance decrease with testosterone therapy. And then on the flip side in blue, all of these things that drive pro-inflammatory antibacterial responses, TNF, IL-6, these types of things, increase. So it's as if your individual's immune system basically shifts focus from one of these processes to the other by testosterone therapy. Go to the next. So what's the implication of this? Well, it tells you that these two processes in our immune system, the type 1 interferon response and the pro-inflammatory TNF response, these are cross-regulated features. It means that if one goes up, the other goes down. We can go to the next, so we don't have to go into all the details. And what we also looked at are which cells in your immune system actually expresses the receptors for these hormones. Which ones can directly sense sex hormones and then regulate our immune systems accordingly. And it turns out that a particular cell type called PDC, plasmacytoid dendritic cell, the main producer of type 1 interferons, an antiviral response cell, has the highest level of expression of the androgen receptor and the estradiol receptor, one of the two, suggesting that these cells are actually sensory cells in our immune system for sex hormones that can regulate immunity accordingly and adapt in relation to hormone milieu. Next. We also looked in public data sets in individuals that do not undergo gender-affirming hormone therapy because we wanted to know if this is sort of an artifact of this unusual situation. But it turns out that in COVID-infected individuals, if you look at type 1 interferon responses in PDCs, monocytes, they're always higher in female cells than males. These are multiple cohorts. You can go to the next. And if we stimulate cells in vitro using various microbial antigens, we see the same thing. We see stronger TNF responses in male monocytes stimulated with TB in vitro in a dish than in female monocytes. Next. So, to sort of summarize what we've learned from this and how this might be relevant to what we are all trying to understand, persistent symptoms after infections, long COVID and other conditions. If our immune system is balancing two types of responses, type 1 interferon responses and TNF, these are responses that would mediate the outcome to an infection with SARS-CoV-2, right? I've already told you that type 1 interferon responses are the ones that limit viral replication, are essential for mild COVID, acute COVID. But they might also increase our risk of long COVID. TNF, on the other hand, gives these pro-inflammatory responses that leads to cytokine storms and severe acute COVID. So these two are cross-regulated, and you can go to the next one. And we think, why is this? From an evolutionary point of view, why would it make sense to balance these two types of responses against one another? Well, evolution only cares about reproductive success and survival, and any trait that would increase the chances of reproductive success would be favored. And what we do know is that TNF, if you look at patients who have IVF treatment and you look for success or failure, you find that elevated TNF levels is associated with poor outcomes. So having high TNF, if you have PCOS, for example, you have elevated TNF, is a source of failure when implantation. Type 1 interferon, on the other hand, is essential because vertically transmitted infections is a critical risk to any pregnancy. So it would make sense, if you're a female of reproductive age, trying to reproduce and have implantation and successful pregnancy, that you want to dampen TNF responses and potentiate type 1 interferon response. And just one more thought before we move on. The 87% or whatever females in long COVID, I don't think it's a coincidence. I think it's a biology and a reflection of these balancing acts that we're talking about. Go to the next. In the male side, why would it be beneficial to boost TNF and dampen type 1 interferons? Well, from an evolutionary point of view, males have always made greater investments into muscle strength and growth, right? While making much less investments into reproduction, if you're a male. Therefore, potentiating TNF might have a really important point, because every time an individual exercises, TNF is released in the muscle and is essential for muscle growth. Type 1 interferon, on the other hand, we know from children with dermatomyositis, for example, with elevated type 1 interferons, that leads to muscle wasting, so weakness. So the opposite adaptation in a male makes sense, that increasing TNF and dampening type 1 interferons. So what I'm trying to tell you is that I think these are evolutionary processes that have nothing to do with SARS-CoV-2 or other, it's to optimize other traits, but as a consequence might impact the risk of developing these chronic conditions. So this was published in September. If you find this interesting, please check out this paper. I'm happy to take any questions. I've worked with a phenomenal group of people in London and Stockholm. Thanks.
Thank you very much. Thank you again for being here. So now I'm trying to show what we have done to translate, basic knowledge in our pediatric cohort. This afternoon we will talk about some therapeutic approaches we are trying. So I've received some funds to study long COVID with no involvement in any phase of our study. So for this initial part, I will go a bit faster. I just wanted to tell you how we are selecting our patients. So since the beginning of the pandemic, we set up a prospective follow-up of all COVID patients, microbiologically confirmed COVID patients that were seen in our hospital. So this allowed us eventually to have a prospective cohort of children that fully recovered from COVID and children that developed long COVID. So this was part of clinical practice, but also research. So briefly, we see all patients at 3 months, if they are fully recovered, meaning they do a normal life, basically. So we will have a lot of discussion about what is long COVID, but a child who's back to playing the activities, who was playing before, going full time to school, going out with friends, doing a normal child, whatever is the definition I will use, is a healthy child and is back, so it's recovered. In those who complain persisting symptoms, we do the most extensive workup we do to exclude at least all the diseases I know that could cause those symptoms. So I know that WHO definition is a bit conflictual on that, but really I wanted to be as much specific as possible. So every patient, first of all, does a workup that can last weeks to months as well to rule out any other possible diagnosis. In those cases, if these symptoms were persisting after this workup and are impairing daily routine, so they are considered as long COVID. So, and this is an important point in my opinion, because sometimes we have parents that come with children saying, okay, my son, he is very tired, and so you ask them, so tell me the day, the week of your son. Yeah, he plays football 4 times a day, he go to English 2 times a week, they play piano. So there is not a case of long COVID, because my definition is a child with persisting symptoms with impact on daily life, and this is also included in the WHO definition. In my opinion, this is a point that is never enough stressed, even in the research papers. It's not just a list of symptoms, but symptoms that severely impair daily life, and we will talk about that, I think, Benita and myself, this afternoon. So basically, very briefly, so far we have followed up about 1,500 children so far. This is the old cohort, not only the long COVID cohort. And what I want to show you very briefly, according to this definition, we had in this cohort about 300 children fulfilling those criteria, even thinking that we may have made some diagnosis wrong, even the 50% of diagnoses were wrong, just pretending. You just compare, in my opinion, this is interesting, the numbers with the multisystem inflammatory syndrome. So everyone in the world has continuously talked about the MIS-C, and in this cohort, these are not all patients, but in this cohort, 5 children had the MIS-C, and everyone in the world is talking about MIS-C. But even if you only consider those patients that have completed the 3-year follow-up and are still symptomatic, these are for sure real long COVID cases, because in 3 years, persisting symptoms of daily life, no other diagnosis, you see the comparison in terms of numbers compared to the multisystem inflammatory syndrome. The symptoms are those that we know: extreme fatigue, so it's not being tired, extreme fatigue, the child is not able to do anything actually. Some children are bedbound like 90% of the day, chronic severe headache, impairing daily life, severe gastrointestinal symptoms, and so on. All the symptoms we know very well. So very briefly, as we mentioned this today a lot, even in our cohort at every time point, the female sex was significantly associated with the risk of developing long COVID. Of course, we don't have only female patients, and particularly those with allergies in our cohort, and those infected with initial variants had a higher risk. But in terms of absolute numbers, we have much more patients infected even with post-Delta variants. It's only that the risk is lower if it's compared with the huge amount of children who got Omicron and post-Omicron infection. So this part is a critical part that is not, I'm going fast here. In terms of vaccination, we found that those children who were vaccinated before getting COVID had a lower risk of developing long COVID overall, but it's not a complete protection against long COVID. We found some unusual opposite evidence in older teenagers. Here, the idea was that we were a bit biased, meaning that only a minority of adolescents got vaccination before long COVID, but then they eventually. We have a biased selection for sure, because we are one of the only centers seeing long COVID patients in Italy. So maybe even the few who were vaccinated and developed long COVID came to our clinic. But so this is perfectly in line with studies from others showing that long COVID vaccination may reduce the risk of getting long COVID, but not entirely protect against it. And this is, in my opinion, another important point. And just a few days ago, another big paper on JAMA was published about the link between COVID and autoimmune disorders. And we have talked before a lot about chronic inflammation after long COVID. In our cohort, children who were infected with the original variant had a significantly higher risk of developing an autoimmune disorders. In this cohort, the prevalence was relatively high if we consider that we don't have perfect data, but this is what is thought to be the overall prevalence of autoimmune disorders in the general population. In our cohort, it was the double of what it was expected. Again, this is just about clinical data, so I'm going fast because I want to focus on more interesting and new data later. And again, we found that the children with long COVID had were at higher risk of having symptomatic COVID infections. This can be either a bias, because those with long COVID can be more prone to be tested by ourselves, but we are having more and more clinical observation and also some studies showing that eventually those patients with long COVID, when get reinfected, had higher probability of having both a symptomatic acute infection, but a worsening of long COVID symptoms. So that is probably, in my opinion, why even children are identifying their reinfections more frequently if they already have long COVID. So now, so I'm moving now more in details about translating evidence from adults to children. And I'm starting with what we know, fatigue and poor resistance exercise was clearly described in adults, so we wanted to replicate this in children, and we did it through a cardiopulmonary exercise testing. I know some patients' associations don't love this test because they can kind of trigger post-exertional malaise for a few days. But when we started everything, long COVID was completely neglected in children. So at that stage of complete neglected the world, we had to even prove ourselves that the fatigue was real. And when it was compared to healthy control groups, all the parameters of the cardiopulmonary exercise testing, meaning muscular functional capacity, cardiopulmonary efficiency, chronotropic competence, and even likelihood of pulmonary vasculopathy were significantly higher expressed in patients with long COVID, in children with long COVID compared to healthy controls, so providing the first piece of the puzzle, in my opinion, that the fatigue complaint in patients, even in children and adolescents with long COVID, was a real phenomenon and not only associated, for example, to pandemic restriction. But then we know historically about post-orthostatic tachycardia syndrome, dysautonomia, and again here we had to start from zero again with the long COVID pandemic, so we wanted to replicate it in pediatric patients. And again here, I will give more details this afternoon when I talk about therapies, but this was the first study. We have more data now demonstrating that the heart rate variability parameters, which are very simple parameters of sympathetic and parasympathetic imbalance, were different in patients with long COVID compared with, in this case, with pre-pandemic healthy control. So second piece, very easy test, 24 hours electrocardiogram with heart rate variability, very easy. Many children have traveled from other European countries to do a 24 hours electrocardiogram in Rome. So this is also a point of thought, in my opinion. But then David mentioned a lot about the neurocognitive problems in children, so it's always easy to say, okay, those neurocognitive problems were due to pandemic restrictions. These were two sisters who were professional piano players, and then they got COVID. They had all the symptoms of fatigue or whatever, and for about 1 year they were both sisters not able to play piano anymore. As they were sisters, the easiest thing was to say, okay, it's psychological because the younger one saw the older one, and the brain PET demonstrated abnormalities when compared to a national database of healthy control routine with brain PET. It was the mesial temporal areas. This paper is being published with abnormal metabolism compared to healthy control groups in areas involved in several complex neurocognitive tasks. But we had also a cluster of children with anosmia or changes in mood. Again, when they were compared to healthy controls, we found consistently, this data are also published, abnormalities in the prefrontal cortex compared to the healthy control database. This was not done systematically because of lack of funding, but in all patients that we have done it, we found similar abnormalities. And so this was kind of another little step in terms of translating some knowledge in pediatric patients. But I think the most interesting part is here. This is a collaboration we did with Professor Palma of Bambino Gesù Children's Hospital. So the preliminary data of this collaboration have been done with Peter Brodin and published on Pediatric Infectious Disease Journal, but it was with a very small control group. So here I wanted to show you directly, as a matter of time, the newer, larger study we have done. So we collected blood samples from a group of more severe long COVID patients. And when I say long COVID, in terms of duration, I know that the cutoff is 3 months, but as I said, we started 3 months, we do a workup to exclude other diagnoses, we have little resources, so it takes months. And so usually these patients are symptomatic for at least 6 months when they arrive in our clinic, but sometimes even more. And we compare with pre-pandemic healthy controls, acute COVID, and the famous multisystem inflammatory syndrome. So we eventually control the group for age and gender. So these are the actual numbers in this preliminary data, which the study is under review right now. Will probably be published in January. So it was relatively quite amazing, I would say, for myself, or even emotional, and this was what Paolo was referring. So if you see the different groups in terms of the principal components of the inflammatory panel only, actually the long COVID patients perfectly cluster very, very well compared, even better than many other groups. So the green are healthy, pre-pandemic healthy controls, so they cluster in a specific way in terms of inflammatory panels. The acute COVID cluster very well, but if you see actually the multisystem inflammatory syndrome, which again has been the most discussed disease in the world, actually cluster less well than long COVID patients. This is not surprising in my opinion, because some missing cases might have been initially wrongly classified, maybe there were Kawasaki disease or the classic pre-COVID hyperinflammatory syndrome, or sometimes severe acute COVID can be similar to BC, but eventually the long COVID patients were really those who cluster better each other, also meaning that we were probably recognizing them relatively well from a clinical perspective. Even when they were corrected in terms of distance from initial infection or age, again, the long COVID group in terms of, again, blood inflammatory markers were clustering very well. So these are some of the top proteins. I will briefly talk about a few of them that differentiated the groups, and particularly the healthy control with long COVID, because it's unusual we have to compare long COVID with healthy controls, but this is the reason is long COVID patients are considered healthy patients from several doctors or family members most of the time. we are very unlucky. I try to be. Okay, I think good that someone does so. So this signature that was selected by Pharma Lab actually was taking a few of those proteins, were very highly accurately recognizing non-COVID patients when compared with healthy control and all the other groups. Of course, this needs to be validated, but this looks something as a very, very, very promising, and in my opinion, reinforce all what we are saying about non-COVID. So if we then selected those patients who had severe fatigue as well, compared to those because we know some patients have mostly neurocognitive cluster like memory issues, brain fog or whatsoever. Those patients with extreme fatigue had a highly differential expression of FGF1, which is a stress-induced myokine, also documented in patients with chronic fatigue, in other patients with chronic fatigue syndrome and long COVID. So again, perfectly in line with what we have from the other literature. But we have more, in my opinion. Just going briefly, the CXCL8 is one of the most important chemokines implicated in activation of leukocytes, regulate endothelial cells, contribute to thromboinflammation, and we've seen how much endothelial inflammation is implicated in disease. Again, higher in non-COVID patients compared to the other groups, actually going with the VC. And this protein has been already documented as differentially expressed in several adult non-COVID cohorts. Or these are the CCL set, which was also differentially expressed in children with long-COVID, again as a role in activating chronic inflammation. And again, this was differentially expressed in patients with long COVID. Or the CXCL1, again, chemotactic for activating T cells. And we've talked about T cells a lot of time this morning, and they come back again in the children somehow. And there are already other studies showing that this chemokine is increased in long COVID patients. Actually, here it has a similar expression as acute COVID and multisystem inflammatory syndrome, which are acute hyperinflammatory disorders that no one questions from in terms of being real disease. And probably last, the OSM. This is another pleiotropic chemokine implicated in chronic inflammation, vascular injury. So again, all the phenomena mentioned this morning in adults seems to come back in our cohort of pediatric long COVID. We are also studying the microRNA profile in children with long COVID compared to pre-pandemic healthy control. So those children recovered. I'll go very briefly here because this is still a very pilot cohort, but we have preliminary data of differential mirin expression, again in long COVID, those lasting a long time, in the whole cohort compared to children who with healthy control. Actually, even those recovered from COVID seems to have a different mirin profile compared to those who never had COVID, suggesting that anyway, even those who recover from COVID, there can be something going on in terms of immunological expression. And this is an hypothesis proposed by Professor Palmer and colleagues. As in this pilot cohort, we are now implementing the enrollment. We found a downregulation of the mirin implicated in this pathway, which again all point toward potential chronic inflammatory pathways in long COVID, as mentioned multiple times this morning from the adult literature. We've talked about microclots. This is another study, again, from the same consortium broadly mentioned as thromboinflammation is being invariably documented in countless other studies. We didn't have the chance to measure microclots, but we could measure platelet activation, which can be an indirect sign of endothelial injury and chronic inflammation. And patients with long COVID, at least a group of them, this was again a pilot cohort, showed a significant baseline hyperactivation when measured with flow cytometry. So again, very objective data, while when they were stimulated, they both responded in the same way. So suggesting again a chronic ongoing stimuli in these patients. This was also in line with the Lyell-Yokers data. So concluding, I really believe, of course, a significant, in absolute number, a huge amount of children are suffering from long COVID from months to years. And in my opinion, all the data suggest that adults and children may share similar immunological pathways, including chronic inflammation and thromboinflammation. Some diagnostics, also when Peter was telling us they were looking for the cohort of long COVID patients with abnormal or objective findings. In my opinion, some tests are very easy to be done and can help selecting those patients, and eventually when they are consistently done with a well-standardized protocol, most of the time, I don't recall actually any long COVID patient that had both these tests normal, and these are very easily done in every pediatric and adolescent clinic. And of course, there is, in my opinion, concrete hope that proteomics can be implemented. This is our goal, to try to implement similar diagnostic tests for recognition, for inclusion in clinical trials, but to expand this not only to long COVID patients, but of course to patients with ME/CFS, and let's not forget that most long COVID patients also have the ME/CFS criteria. And I will conclude with that. I will present all the team involved in this in my afternoon presentation. So for now, thank you, and I will give the talk to Leah for the second time.
Okay. So it's my pleasure to introduce Leah Nusker, who is actually co-director of Massachusetts General Hospital, which is the Tuberculosis Center. And I'm going to change presentation. Okay. So Leah, it's a pleasure to have you here. Thank you.
Thank you so much. Thanks to Daniel for putting this conference together and inviting us all here. So I would say that, I did a search of how many long COVID studies there are in adults. There are tens of thousands. How many are there in kids? Handfuls, and half of them are from Danilo, and he's just been really a machine. So this is fantastic that you're really pushing things forward. So, this talk is about covering what we've been learning from adults and how it translates to kids. I sort of took it a sidestep a little bit and thought about PASC generally, or post-acute sequelae of COVID generally, because I did do a lot of work looking at multi-system inflammatory syndrome in children, and that being the most severe version of post-COVID syndromes, and that we can actually learn a lot. And so just to sort of give this visual, I think that for acute COVID, there's a very big difference in how children respond in that acute infection. Age is the greatest risk factor for death for some severe morbidity in the acute infection. And then after that, we learned that kids can develop this severe post-acute sequelae, MIS-C or PIMS. And then, and again, this was in children, so again, some stratification between adults and kids. And now we have long COVID, and there's still this misconception that there's a big difference in what we're seeing. And I actually don't think there's a big difference. I think the pathology seems to be similar based on the work that others have been doing and that we've been doing. And so, just something to keep in mind, and it's something that I think we need to press on, help inform clinicians about to keep an open mind for kids they suffer from long COVID too. Preaching to the choir. But to think about PASC, the spectrum of PASC. There's similarities between MIS-C and long COVID and the multi-system and organ involvement. MIS-C is a lot more severe. Kids have shock and myocarditis. But long COVID also has that endovascular phenotype with POTS, symptoms of lightheadedness, this and dizziness. There's GI symptoms involved, neurologic symptoms involved, and sort of general symptoms too. And so what we showed, and Peter kind of showed earlier, is that in MIS-C, after you've been exposed to the post-COVID syndrome infection, after you've been exposed to SARS-CoV-2, weeks to months later, you still have SARS-CoV-2 in your gut. And we showed that there was positive RNA in the stool. We showed that there's increased zonulin released in MIS-C, and that is a regulator of tight junctions in the gut. And so that signified that there was a breakdown of those tight junctions of a leaky gut. And we also showed that viral particles were leaking into the bloodstream, and we were detecting the spike protein in the bloodstream of these kids. And that correlated very strongly with this very inflammatory response. But what others have been showing over time, this data was shown earlier, is that many studies actually have shown that SARS-CoV-2 persists in the gut. This was not shown earlier, but there's increase in zonulin release in long COVID. So again, there's that marker of this leaky gut in long COVID. And there's also spike proteins in the blood, and this was detected. There's been a few studies that have been coming out over time as well. So there seems to be this same sort of sequential march. And so what we've been looking at is what does this mean? Sort of taking it one step farther. If you have spike in the blood, what does that mean for your inflammation? And again, we're going to look back at what we've learned from MIS-C, and we've been working on some things in parallel. So at MIS-C, we did this intensive profiling where we looked at kids with acute COVID, MIS-C, and their healthy controls. We really did some extensive profiling of their neutrophils. And we saw that when there's spike in the blood, spike forms immune complexes, and these immune complexes activate neutrophils to release NATs, or neutrophil extracellular traps, and also sets off this very inflammatory pathway. And so again, this is just the data that we published looking at MIS-C. I'm going to show some videos in a second, but there's very elevated NETosis, or these extracellular traps. And there's also markers of cell-free DNA are increased, and that's another marker of NETs. And then other sort of myeloproxidase marker of NETs. And here's some videos. So in the top video, our healthy controls, you put them on the plate, and a couple of them died and turned green. So here's just looking at a light microscope. Here is a dye to look at DNA, so you can only see it when there's a breakdown of the cell membrane, when it either dies, it sees like a tight circle, or when a neutrophil cell, the net is released, it forms more like a puffy tennis ball, or sort of spreads out. And then here's just the overlap. So there were 3 cells that died. They stayed nice, tight little cells, but they were just cells that really very little NETosis. And in the bottom, we're going to see just neutrophils from kids with MIS-C just put on a plate. We didn't stimulate them in any way. And you can see all of these start popping out, and they fade away because the DNA is spreading. And so marked difference between the two. And then we looked one step further and wondered what was triggering this. And again, going back, it seems as though the antigen, so the spike in the blood, when it binds to antibodies, it forms immune complexes, and those stimulate NETs. And what we're seeing is we can artificially do this by adding plasma and spike and adding it to neutrophils. And but it's comparable to what we're seeing in MIS-C. We also could deplete the immune complexes by either IgA or IgG and see what's really driving this. And it was the IgA immune complexes that really seem to be driving MIS-C. So now we're doing this in long COVID, trying to understand the spectrum of inflammation. This is work in progress that hopefully we'll be submitting for review soon. But we have 21 kids with long COVID where we have fresh neutrophils for profiling. We had 19 kids with from MIS-C who we're using to compare again to see the spectrum of CASC. And then we have 60 controls. And again, what we're seeing is remarkable neutrophil extracellular trap formations. And here's just an example. And you can see in this smaller cohort that the amount of, actually here's sort of two combined groups. We differentiated long COVID who are spike positive and spike negative. And they really have elevated neutrophil extracellular traps almost to the same degree as we're seeing in MIS-C. Pretty comparable actually. And same thing with the extracellular DNA. And so then we're thinking, well, maybe there's, the spike, it's a small percentage of people with long COVID, 20 to 40% based on different recent published numbers. But we wanted to see maybe there was some dose-related, response. Maybe in MIS-C there's a lot of antigen that you're getting all at once, whereas in long COVID it's a little more indolent. The spike can come and go over time based on what others have found. And so we do see a dose-related curve. So the more immune complex that you have, the higher neutrophil activation and sort of, and then we looked at cytokines that correlate with these as well. And I'm just showing the spike positive as compared to the MIS-C. These are, the color coding is normalized to healthy controls. So white is that they're the same as controls. Red means those cytokine levels are elevated. MIS-C is a very inflammatory disease. There's a cytokine storm, so there's lots of reds and a lot of elevation compared to healthy controls. But sort of remarkable that the long COVID patients also have these elevated inflammatory response, and there seems to be some patterning we're starting to see. So how can we treat PASC? I want to sort of talk about some of the efforts that we're doing really quickly. I know we've got a lot to get through, including some time for questions, but I want to share some recent data too. So we're looking at this leaky gut, and in my mind, there are 3 different ways of targeting long COVID. There's targeting the reservoir, right? So people tried Paxlovid in large cohorts. The large cohort it didn't work, and David already pointed out some of the things that we need to consider, but that's an important strategy. The second is targeting this leaky gut. So sort of creating, if you're going to have a reservoir, maybe you can contain it in the gut if that's where in fact the reservoir is. And so that's a strategy that we're looking at. And the third one, which has a lot of interest and activity in adult patients, is targeting the immune response. So the microclots, the monoclonal, the hyperactivated T cells and B cells. And so you can sort of see that maybe there's probably a multi-pronged strategy that needs to be done to target the different places. So lorazepam is the treatment that we're using to target this leaky gut. It regulates tight junctions. It helps restore tight junctions. Here's just an overview. I'm going to skip this time. But it's very safe too, which is reassuring. It's very safe. And so it is an oral medicine that is not absorbed into the bloodstream. And so it has very limited side effects because of that. And it's been extensively studied, often and most commonly in celiac disease. And so when we look at a cohort of kids with MIS-C, so again, learning from what we've done in MIS-C, antigen levels are high, and you treat them with, here's all of the antigen, spike, S1, nucleocapsid. Many had detectable levels. You treat them with IVIG or steroids, it actually did nothing to change their antigen levels. This is our first publication showing the impact of lorazepam. We had a very severe patient who was hospitalized. For acute COVID. He was in the hospital for a while and then developed MIS-C. And we have a few things we looked at: CRP, but most importantly, and other inflammatory markers and fever. But here's the red is the spike. So when we treated with IVIG and steroids, you saw the inflammatory markers go down, but they came back. And we saw the spike levels were persistently high. Then we treated with lorazepam, and there was a remarkable drop in the spike levels in the blood, and that corresponded with a drop in inflammatory markers. So again, this was the first proof of concept that maybe targeting this spike leak was helpful. We did an open label trial where we gave 4 patients lorazepam and just retrospectively compared them to those who were not. And even in this open label group, we did see faster resolution of GI symptoms and faster clearance of spike. But that was limited by being open label, so we were a little biased, essentially. So we just completed a phase 2 randomized placebo-controlled trial where we were blinded as to who was treated with lorazepam versus placebo, in addition to whatever their standard MIS-C treatments were. And we had 6, it was, admittedly a small trial still because MIS-C was rare, even more rare now. 6 treated and 6 with placebo. And we saw significant improvements. We met our primary endpoint, which was resolution of GI symptoms, up here, resolution of GI symptoms within the 3 weeks of the treatment that we had. And the people who were treated with lorazepam had significantly faster clearance of symptoms by day 7 and day 14, equaled out by day 21. We looked across all of the different types of GI symptoms. There was a significant improvement. And we asked the question in many different ways. And so it really reassured us that there was an improvement. And again, you can sort of see that the coloring is not great here, but it showed that there was clustering. And so again, reinforced that what we were seeing was real. We also looked at spike clearance over this time. And this is these are presented great, but here you can see the red, this is time to spike clearance in the blood. And those treated with lorazepam cleared the spike more quickly. And when we looked at each time point again, you saw significant clearance. And the placebo group, in fact, some of them didn't even fully clear the spike antigen by the end of treatment. Interestingly as well, we saw, I'm going to skip it for the sake of time, but there was some antibody data that was sort of interesting as well. And importantly, they were improved functionally. So they were kids who were treated with lorazepam were able to return to their usual health and their usual activities faster. So this is all very encouraging. And now we're running this in long COVID. This is a study that is ongoing. We've expanded. It was initially just children, but we want to move the study faster. So we have expanded to individuals through age 50. The one caveat is because we are specifically looking for spike clearance and as a marker for improvement, people have to screen positive for spike in their blood. So as part of screening, we do look for spike. And then we treat in a 2-to-1 randomization ratio, and we are looking to see if people will improve over a 3-week course. And we have some different long COVID specific outcomes we're looking at. So in summary, I think there's a lot that we can learn from MIS-C. I think we need to work harder to phenotype long COVID in children because there are parallels that we are seeing, and they need to really be highlighted more. And because there are these parallels, I think pediatric populations need to be included in clinical advances. And so there's no reason that treatments like rapamycin, IVIG can't be used in children. They're not novel therapies that are being used, and we know that there's similar pathology. So I would strongly encourage expansion of clinical trials to offer something to these kids. With that, I just want to say thank you.
Yeah, thank you very much.
Okay, that's better. And thanks in particular to Danilo, but also in part to Danielle. It feels a bit like payback time because when we started looking at incorporating long COVID into the global burden of disease study, we quickly realized that published studies were useless. We couldn't get enough information out of it. And so we put out a call to a whole bunch of researchers that we knew were sitting on follow-up data. And we got fantastic response, including from Danilo and Daniel. So thanks very much for that. So when we started this, it was under the principle of the global burden of disease that we want to quantify all sources of health loss globally. And so when this new concept arose that there is a serious post-viral syndrome for SARS-CoV-2, we said we have to tackle this. And we concentrated on the types of symptoms that aren't easily captured by any other disease estimates. And so we decided, instead of having instead of well, I've lost my train of thought here. But so we decided to concentrate on three symptom clusters that were very commonly reported in that emerging literature. It was also a little opportunistic because those were things for which we had disability weights, which are key to quantifying health loss in the Global Burden of Disease study, where we can then make what we call years lived with disability, that are the non-fatal component of disability-adjusted life years. And that's the common metric in which we quantify the health loss from all conditions. And so we chose symptom clusters around ongoing respiratory symptoms, shortness of breath being the key symptom. Then a cluster of fatigue with bodily pain and mood swings. And that's a consequence of disease that we previously were already quantifying post-dengue, post-Zika, post-Ebola. And then it's a third cluster with cognitive symptoms like memory loss and problems with concentration. So these are these disability weights where they range from 0 to 1, and 0 is perfect health and 1 is perfect ill health, if you like. And so you can see the values here for different levels of respiratory symptoms mirrored on the disability weights that we use for COPD. The cognitive ones are mirrored on mild and moderate dementia. We did not use severe dementia because that seemed out of range for the kind of symptoms associated with long COVID. And then this post-viral fatigue syndrome with its own disability rate. Next is, so as I said, extremely low yield from a systematic review. Because it largely was counting symptoms or where it was reporting the single symptoms, it didn't quite fit with the constructs that we had. We turned to two large sources of administrative data, the private health service database that we were able to analyze. And we worked with Ziad Al-Ali, who has done extensive work on the Veterans Administration data in the US. Big advantage, enormous numbers. You can clearly identify cases and controls, but a big drawback is you have to rely on reports of symptoms through ICD codes, and we know that it's very deficient. So it's not very specific, but with the large numbers, it does help to see patterns over time. But the mainstay was, as I mentioned, collaborating with people doing follow-up studies. And the way we did that was by taking all the instruments used in these cohorts, often very different, and trying to harmonize algorithms that would most closely approximate the construct that we were trying to quantify. And only 4 of these explicitly had questions for each of the symptoms. Yes, you're acknowledging now this level of fatigue, but how was it before you had SARS-CoV-2? And that's important because there's a big danger of overestimation in this field, because all these symptoms are extremely common in the general population. And so you need to look at the delta between people who had infection and people who did not. So this is the distribution that we found with the still limited information we had in 2021. And roughly it affected 1 in 10 women, 1 in 20 men, and 1 in 40 children, we said at the time. And here you can see the contribution of each of those clusters, and also how these clusters of symptoms overlap in individual patients. An overall disability rate of 0.23, that probably doesn't say much to most of you, but that's relatively severe. That's as bad as inflammatory bowel disease, or severe neck pain, or complete deafness. So it's on average very considerable health loss. And the poor individuals with overlap of all three clusters, that quickly multiplies to a very high level of disability. So we found at the time 145 million new cases in the first 2 years of the pandemic, 90% coming from non-hospitalized cases, so more mild community infections, a predominance of women, ratio of almost 2 to 1, and at the time we said at 12 months post-infection, 15% of cases had not remitted. But that was a relatively bold statement because we only had one study going out 12 months at the time. So, in an update that we're about to finalize and that will slot into the Global Burden of Disease 2023 results, which are due to be published early next year. Many more studies, including studies specifically among children. A decent number of studies following up community infections. It's easier to study people after a hospitalization, so there are more of those kind of studies. And importantly, a number of studies with longer follow-up, including a couple of studies with over 3 years of follow-up. Now, this is something very new that we only recently started looking at. These are the results of 7 studies that compared the risk of long COVID, no matter how they define it within a study, between variants. And the majority of these cases were PCR confirmed and sequenced, so that they knew which variant had caused the infection. And what you see here is, even though these studies report very different levels of disease, can I point with this somehow? Anyway, you can see the green blobs at the bottom there. That's a Swedish study that relied on GP diagnoses. And so they have reported very low levels of long COVID. But we think that's probably quite an underestimate. And so when you just take the studies as they are, they're all over the place. Once you put in statistically random effects on each study, you see that, and you multiply the random effect with each of the data points, and you can see that they much more closely follow a very similar pattern. So the green ones over time show very similar results as we see from these other studies. The conclusion from this is that there's a big gradient over time dependent on the type of virus, in the risk of developing long COVID. Much, much higher with the wild virus, and then diminishing over alpha to delta to Omicron. This extends out to the middle of 2022. What's happening after that, we're of course not sure. What we're doing with our current estimates is assuming that this wonderful curve that we've drawn will continue to follow that trajectory. Then, so that was about the risk depending on when you pick up the SARS-CoV-2 and what variant was dominant at the time. These are follow-up studies that start with identifying cases and then over time look at how long these symptoms are persisting. This is a graph that we've made for those under 20 in community cases. And you can see that there are quite a lot of different studies that contribute to this. And this then says, okay, this is the initial level of disease. Over time, people get better. And what is there for the amount of health loss over that trajectory of recovery that we then capture with these disability weights in our metric of values. For hospitalized cases and ICU cases, we had far fewer information for kids, and so we ran models with adults and children combined. And you can see in these that for more severe cases, that the initial risk is greater, even more in ICU cases, where you can argue, well, there's an overlay of what's also known as a post-ICU syndrome with very similar sort of symptomatology as with long COVID. But that they follow a relatively similar pattern of recovery over time. What these new data tell us is that we overestimated the pattern of recovery because we had so little information going out long enough to inform that. That instead of a relatively rapid recovery and 15% of cases after a year still having symptoms, we're now coming up with quite a bit longer durations, having that much more information to inform this, and therefore you get quite a different build-up of estimates because the incident cases stay a prevalent case for much longer and therefore accrue many more DALYs than we originally had estimated. The decline in the risk, depending on variant and time period and whatever influence of past infections and vaccination, leads us to make quite different estimates on the amount of disease and the amount of incident cases of these different symptom clusters. You can see quite a drop already from those who got their long COVID in the middle of 2020 to just 1 year later. And those estimates drop even further if you go further out into the pandemic. These are currently our estimates of the proportions who are still a case at 12 months. And you can see that it's more in the order of half of the cases not yet having recovered, or 40-50% not having recovered, as opposed to the more optimistic information that we put out earlier. What does it mean in terms of the total numbers globally that we estimate? And even though we have limited geographical spread, though it's increasing of studies that have followed up people with long COVID symptoms, we do see that wherever people look at it, it is there. So it's not something that's restricted to the countries from which we've been able to use the data, we believe. And what you can see here is that, so the middle column is, if you like, the incidence of long COVID relative to our best estimate of the number of infections that have happened in each of those 5 years. And you can see a very large number of new cases because of that much higher risk with the wild virus that was circulating, and that's dropping down. And then if we extrapolate, you can see dropping down to very low levels that we expect in 2024, despite all these billions of infections in the world. But there's a delayed effect on prevalence. And so you see a big buildup of prevalence towards 2021, 2022. And then we're estimating that these numbers are coming down quite rapidly, but with a lot of uncertainty of, is this really what is going on, because we don't have enough information yet. Translating this into a ranking of causes of non-fatal health loss. Our number one cause of non-fatal health loss in the global burden of disease is low back pain. In 2021, we haven't yet fully calculated through to a ranking list. I suspect long COVID will rank very close, if not a little bit higher than low back pain. So very significant impact on the lives of people. But with, if we are right, a much more optimistic story about it having dropped in significance in terms of ranking against all other conditions in more recent years. These are the numbers for kids. Quite a bit smaller because of the lower risk we estimate in children relative to adults, and then again men against women. But still very significant numbers. But the same pattern where you see the greatest number of new cases in 2020, and then dropping quite precipitously if indeed we believe that our drawing of the curve continues to occur into 2023 and 2024. So compared to what we've published as part of our Global Burden of Disease 2021 results, we have now incorporated this notion of a much higher risk early on in the pandemic, when there were, and we also toned down on our estimates of the total number of infections, particularly in the first couple of years globally. However, because we're estimating a much longer duration, our estimates of valleys for long COVID are going to go up quite considerably. The average disability rate is pushed up a tiny little bit, but as I said, it is significant. And of course, this is an average. And someone who trained me during a Masters of Epidemiology said, you can have your head in an oven and your feet in the freezer, and on average you're all right. Well, here 0.23 on average master, there are quite a few people with much lower levels of disability, but also a considerable number of people who are really, really out of whack.
Добрый день, мне очень приятно быть здесь, я рад вас всех видеть. И я надеюсь, что вам понравится мое выступление. Oh, sorry, yeah, you didn't understand me. I'm really sorry. But that's exactly what happens in research when we study different things. Right? We are immunologists and clinicians and researchers and patients, and all of us, we have different views, and we have ego, and we have our research, and we have somebody else's research. And we need to try and find the common language, like English is current accepted English in international research. So I don't have any conflict of interest whatsoever. I don't receive any funding with regards to long COVID, and never received, apart from a small grant from British Embassy in Moscow, which were very kind to help us with our research. Something I wanted to show you is this map. And these are greatest empires that have ever been created in this planet. British Empire and Russian Empire, and so British Empire, Mongol Empire, Russian Empire, the kingdom. I just took the top four, the most prevalent. So a storyline of power that been ruling the world. So all of us, we can have our own opinions about this, but we need to admit, we need to accept that the power and money to a certain degree impacts the world. And we should not forget this. And we should not forget the story of Theranos and Elizabeth Holmes, because it's a very, very interesting story, and most of you, I guess, know it. If not, I would highly recommend looking, reading, watching. This all started with very good intentions, great intentions. She really, I believe that she believed in what she was doing at the beginning, and then with good intentions, it came up as one of the biggest fraud in the history of everything, I guess. And if you look at the faculty panel of this meeting, which I'm a part of as well, it is quite Anglo-centric, isn't it? But again, for a reason, right? Because this is where research is coming from, this is where funding and resources are available. So we need to keep this in mind, to bear this in mind. And this is our life. I'm sorry I stole this from a good friend of mine, shared this slide with me, a cynical slide of academic research, because that's what happens over and over again. We do research, publish papers, get grants. And we publish papers to get grants and grants, get grants to get going, right? So that's a vicious circle. Something I will be talking about is boring methodology and standardization, and the outcome standardization, as well as other things. I would like to begin with long COVID. This term, as you know, has been invented by the patient community, which is wonderful, and it's great, and it's an important step, and this is one of these rare examples when we see that the patient community really drove us where we are now, because we're here now today because of Danilo, and because of our patient colleagues that invented the terminology and brought attention to the problem. However, throughout my talk, I would like to try to pass my personal views that I think we need to try and slightly move away from the initial term of long COVID, or at least reach an agreement and possibly harmonize the terminology. So this is a post-COVID-19 condition, as you know, developed by the WHO. I know that this terminology has been heavily criticized. But what they tried to do is, obviously comes from very good intention, they tried to harmonize and define something that has not been defined previously. I will leave aside, I will keep aside the political side of it, so the development of the term that would be distinct, term that been invented by, that been developed by the WHO, etc. But it says that it's, as all of you know, a confirmed probable SARS-CoV-2 infection, in people experiencing symptoms that last at least 2 months and occurred within 3 months of acute COVID. But there are other definitions, and people invested their time, and funders invested a lot of money in the development of these projects. So, of course, they also there's some geographical predisposition to use one or another term. So, as you know, and sorry, there's a wrong reference here. It should be a JAMA paper, obviously, from colleagues from the US that used this symptomatology-based approach in providing definition of PUSC. But recent endeavor, which I'm sure all of you are aware of, in the US, tried to develop long COVID definition, right? So is it the same long COVID as patient defined at the very beginning, or is it a different long COVID? So would patient community agree that this term is equal to what they meant originally? I guess it's more a question to our patient partners. But that's, again, long COVID is an infection, chronic condition that occurs after SARS-CoV-2 infection presented for at least 3 months. So 3 months is this threshold that's been selected in this definition. And if you go across the definitions, and I think they beautifully collated this table in the initiative, there are still discrepancies, right? Because you see that some definition they align with term long COVID, others don't, right? We have attribution to infection, so some of them they say that it will include a symptomatic mild and severe COVID, so UK-NICE doesn't have it, SOH doesn't have it, WHO children does not have it, right? So there are differences in definitions. So I'm bringing attention to this because, yeah, that's a small thing, but we're defining this in our studies. Most importantly, think of this will be definitions used in clinical trials for subgroups of patients that will be targeted with treatment. So there is timing in some definitions, it is present, in others it's not. And clinical features, as you can see, there's a lot of discrepancy as well as patient-oriented features, as you can see, some of them include risk factors and others don't. And I think Danilo made a very, very important point about symptomatology making an impact on daily functioning. Because there's so many studies that have been showing that symptoms occurring, persistent symptoms after COVID. So are we talking about long COVID, or are we talking about something else? So I've been leading a number of cohort studies back in Moscow, and this is one of the first studies that have been published in pediatric population. So our study, we managed to record symptoms over time. As you can see, symptomatology, something that we see across the board on over 500 children, and we also looked at some risk factors with elder children and some possibly links with allergic diseases, although it's not very robust. It's based on symptoms. It's just based on symptoms, although we've seen a very, very nice decline over time, suggesting that possibly the children recover over the period of time. But what we did then is, and this is aligns very well with what Theo was talking about, we ran another cohort at the time of Omicron, and at the time of Omicron we were fortunate enough because in Russia many worried parents, they bring their children with common flu, with common cold to the hospital and say, oh, my child is dying, it's a fever of 38.5, that's it, please admit them to the hospital. So, and for the sake of, I don't know, it's difficult to explain why, but many of these children are getting admitted to the hospital, hundreds of these children. So we used this opportunity and we looked at the time of Omicron at hospitalized children with Omicron, PCR positive, express test positive, versus PCR negative, express test negative children with common cold, and there was absolutely zero difference 6 months later. no difference whatsoever in any of the symptoms. We use the same questionnaire that been used in our previous studies. If you compare it with previous variants, there's a dramatic difference both between those children that been exposed from our cohort that I demonstrated on the previous slide, from initial variant and reference group and initial variant and Omicron, initial variants and reference group and initial variants and Omicron variants. So there is something there. As you can see, there are dramatic changes and fatigue that is commonly reported. One of the major symptoms, obviously, has been also different to a substantial degree between the groups. So this is very well aligned with what Theo just been showing. But not just this. We're talking about long COVID, but look at this wonderful graph. So you can see patients and control subjects. So there's a huge level of fatigue, much larger incidence and controls and weakness and dizziness, difficulty lifting things. And there are some other activities, as you can see, so tremendous impact on quality of life. But it's not long COVID. It's botulism 4 years after infection. So this is something that I choose the very dramatic example, and that's actually the only very well conducted study on botulism with controls. This is something that you see in botulism. You see it in other infections, as you know. You see it in dengue, for instance. You see post-dengue syndrome, and again, I selected this study because it has control groups. You have dengue patients and you have people after febrile illness, and still you see individuals who are fatigued, and there's a substantial number. And it's very important to remember this, in my view, because we're talking here not just about long COVID, we're talking about a much broader category, about post-viral illnesses. And we've been talking about this for decades. It's just that COVID instigated new interest in the subject. So this study been published almost 20 years ago in the BMJ, and it's an Australian study that's been looking at individuals. And as you can see, they're saying what is already known on the topic of post-infection fatigue syndrome. That means diagnostic criteria of fatigue syndrome may follow abstain virus infection, etc., but not common viral infections. But what authors found in this study is that 12% of participants at 6 months had different disabling sequelae. So again, we're not talking about long COVID here. We're not talking about SARS-CoV-2. We're talking about different viruses and viral infections, and all of them resulted in a similar degree of resolution of the symptoms over time. And even earlier than that, in 1994, there were studies that were on post-fatigue syndrome, post-viral fatigue syndrome, so colleagues were trying to bring this attention for years to the problem. I think it was beautifully summarized in this paper from Akiko Iwasaki lab, where they outlined a large number of different post-viral conditions, post-Ebola syndrome, post-chikungunya, acute fever fatigue syndrome. This is very well known. So maybe it's a time for us to try and bring efforts together to study not just long COVID, but to study post-viral illnesses. And this is important because when we're looking, I already mentioned clinical trials. When we're moving to the clinical trials, we're looking at the outcomes, at the endpoints that we need to address. And in order to address these endpoints, we need to select them. And we can select hospitalization or some objective measurements like lung function, test the biological markers, or needs and rescue medication, quality of life, morbidity and mortality, symptoms. There are so many different things that we can select from. And we can measure these things in a multitude of different ways using multiple scales. And examples referring to other fields demonstrate how people were using thousands of instruments over the years. And that's something that we do and something that happens, and sadly results in problems. And I don't want to bring an additional layer of complexity here, but I will. This is a meta-analysis on treatment of pain in fibromyalgia, which demonstrates, if you look, all of you are very familiar with reading meta-analysis, it demonstrates an effect of an intervention. So you see how effective intervention is. However, you see this dotted line, and this dotted line means minimal clinically important difference. So something that, well, actually, the numbers that we see on the scale, even if it's statistically significantly different, whether it really reflects whether the patient feels it, whether the patient feels the difference. And if you look at this meta-analysis, no, patient doesn't feel the difference, although statistical difference is there. And this is very important. And same sadly applies to biological markers, because we've seen beautiful presentations, and again, I'm not trying to tackle what being presented, it's just for us to remember that this should all be taken in, so that methodological limitation should be taken into consideration. Very often, first of all, in biological markers, we're talking about association, and we don't know whether the statistical significant difference is biologically meaningful. So this is not certain very often. And secondly, the success rate, I'm just using again, thanks to my friend, all of those who passed this slide to me, a success rate of colon cancer biomarkers with thousands of publications, over almost 2,500 biological markers are identified and tested, and that's what people continue doing. And only 3 diagnostic biomarkers that are used clinically. So again, just for us to remember not to get into the same potential pitfall. So I'll just skip a couple of slides. So, and just to remember that obviously depending on the outcomes, policymakers will make decisions because they will value the outcomes much higher if they are more relevant or more disabling, like there was demonstrating the weight. So in order to harmonize, we run core outcome set processes trying to reach an agreement on the most important, critically important outcomes to be measured and the optimal ways on how they should be measured. And this doesn't mean that other outcomes are not important, it's just unrealistic to measure everything. That's why we've developed a couple of core outcome sets. Again, I won't stop on the process just to show you, so you can look at the publications in the Lancet Respiratory that's on adults, the PCCOS project. And then we continued into PCCOS Children project, where we developed the core outcome set for pediatric population. Many of you been involved in this, so again, I don't need to stop on this in great detail. Those that are unaware may have a quick look. But I think that this is again very important. The selection of outcomes and the reporting of outcomes is very important, and we need to bear in mind that clinical trials, particularly in such a heterogeneous condition as long COVID, will be focusing on different aspects. So it's important to measure outcomes in a similar fashion and using similar instruments, because otherwise we'll get up into a nightmare situation where we can't compare the data. So in the last thing I just wanted to, again, because this is more, as you can see, talk about just trying to bring attention to methodological problems. It's all due to our perception. Many things are due to our perception. Same applies to data analysis. So if you look at this picture, some of you may see a lady who's kneeling and praying, and others may see men with mustache and beard, right? Possibly some of you see both, I don't know, maybe some of you see both. So same applies to data. People see data differently. As you can see, people see outcomes different. People see pathology different. Everything that can be seen different. And there was a couple of beautiful publications. One of them being published about 10 years ago. What colleagues from Germany did, they provided 29 research teams with the same data set. Just I'd like to reiterate, the same data set that had data on football players from around European leagues. And they posed a question, could you please test a hypothesis whether a skin color of a football player is associated with the risk of getting a red card? And as you can see, they got dramatically different results. Some more some similar, others very different. The same data set, we're talking about prevalence, and Theo was reflecting on the heterogeneity of the data and problems with the data. Here we're talking about the same data set, they all had the same data set and they came to different conclusions. And you will tell me, yeah, it's not relevant, it's just football, who cares? Well, I do care, I like football. But I can give you an example. The paper that had been published in Nature just a few years ago, which is very relevant to us on the medical data sets, so neuroimaging. So they looked at the variability of neuroimaging results across different analysis pipelines. So they provided the same MRI data set to independent analysis groups globally and requested to test 9 predefined hypotheses. So again, this data set is publicly available if you're interested. And again, cautious of time, I'll just skip to the next slide. Very similar to the previous results, as you can see in football players. Football players, MRI, it doesn't matter, it's the data. So people analyze differently. Something that I was particularly impressed with is that, as you can see, that there was a significant variability, and the tests, the approaches were very different, and the analytical choices significantly affected results. And this line, I think, is the most important. Analysis pipelines were highly individualized, with no two teams using identical workflows. Right? So we have heterogeneity in the terminology, we have heterogeneity in the outcome selection, as well as statistical analysis. So I hope that it was useful, and I think that the take-home messages, at least how I see this, is that there are still several different definitions, and we need to find a way to reach an agreement, because I still review papers and I see that people define long COVID in thousands of different ways, although they call this long COVID. We need to develop possibly a classification of post-viral sequelae to try to bring it together, make it more robust, refined, and harmonized. And the overarching research of post-viral sequelae was really urgently needed, because we compare people with long COVID, very selected population that end up in the hospital with healthy controls, but not with people, mainly, I'm talking, there are some exceptions, obviously, there are wonderful publications where they compared with other post-viral infection sufferers, but we need more of this, because there's still people studying post-dengue, long COVID, and post-other viral syndromes quite separated, so we need to bring them together. And outcome conversation, obviously, unification of terminology is required, and I think that guidance on a more unified approach to data analysis may also be helpful. We're kicking off a study very similar to what I just showed you, with one data set and many analysts, and Danilo and Terence have already kindly agreed to participate, and if any of you are interested, please join us in this process. So yeah, I'm sorry if I went slightly over the time.
For my presentation. So of course, I am the first one who was trial for long COVID in children in Europe, and even more in Italy. We are really struggling to have resources to do so. Personally, I've applied several times. The last time, I'm saying that because maybe some of you might be called as a reviewer for my last application, so I clearly tell you. So I applied 1 month ago to the ERC starting grant for a trial, including also anticoagulants. So in case you receive this application, maybe you can know what to say. Okay. Clearly, so one is pending, one more we are presenting, and the other thing, you heard that, but I want to stress this again, because the actual paper, the list of symptoms in papers, do not fully provide a picture of what is a typical child with long COVID. Is really, literally desperately ill, and as such as the family. This is just one of the several one. He was perfectly fit, Nicola, 14 years old, one of the best at school. Got COVID the second time during Omicron this time. So again, I want to stress that compared to original variants, the risk can be lower. But if you consider the large number of the denominators, people are still having a long COVID, even with reinfection. So at 14 years of age, from a perfectly super active life, he was a football player. He was playing on the side, so one of those players running most. And since then, he was not able to finish without being tired and destroyed one floor of stairs. So he left sports. He stopped going out with friends. He started having memory issues. He started struggling to needing much more efforts to study and to remember the things he was used to study in some hours, for example. And so he started crashing while trying to push himself, and slowly he was unable to attend full-time school. So this is a very classical situation. I actually have children or young adults since a while stating some patients had cancer as well. So they told me when I had cancer, at least I had some chance, some hope, something to try. And we had, I think each of us, young people that either literally took their own life. Some patients also suicided here in this hospital, and we are only after some doctors recognized that were perfectly healthy, got COVID, got depression, and took their own lives. And other young adolescents as well, thinking about, or at least having some of these thoughts. So, of course, here there is a very difficult question. So, wait and see, or let's see what we can do according to the latest literature. So, this was the question we asked ourselves. Initially, independently from what were the chances to prescribe or not in our setting. So, initially we said, okay, let's follow these children and let's see what the literature will tell us month by month, for example. This is, I think, another misunderstanding applied in long COVID. You know, the do not harm from like 3,000 or more years ago. Is it still valid nowadays when we have several similar conditions and we do have drugs for which we have decades of experiences in pediatrics? So I don't know. My personal answer was, again, let's see if we can do, and of course we adapt month by month, of course. And a very last reminder, for us as a pediatrician, and even more for neonatologists, most of the things we do are off-label, actually. So for several multi-drug resistant bacteria, so we are in, so 95% of my job is general pediatrics and pediatric infectious diseases, because I don't have a formal long COVID or chronic fatigue clinic. So all the multi-drug resistant pandemic is about off-label use of new antibiotics in pediatric population or in neonates, because these drugs have never been trialed in children. So personally, we adapted a step-by-step approach. We also have some similarities. So first of all, we start with a very simple 10 minutes 10 tests. I agree with what Benita said. Sometimes we are not that rigorous with the cutoff when we have the signs and symptoms along with the 10 minutes 10 test. But every child does a 24-hour electrocardiogram with heart rate variability and the cardiopulmonary exercise testing, as I said. The stent test is quite obvious, but in my opinion, a simple 24-hour electrocardiogram is even simpler. So this is, for example, a child admitted in the hospital. They cannot go to the bar, for example. So they can just stand up, go into the toilet, and maybe working some meters along the ward. So and you can see how the heart rate drops up and high, with very minimal efforts. It's like recording multiple stem tests during some hours. So this is really a mandatory test, and children literally come from several countries in Europe to Rome to do this test, which is maybe 20 euros test. So how do we treat them? The ivabradine, no, I don't want to say anything about ivabradine, but it's more a kind of parasympathetic-like activity, and we do have some responders and some non-responders. Probably we are understanding with the 24 hours, with the heart rate variability, why some children respond or not to the ivabradine. So as I said, in our previous study, we demonstrated differences in heart rate variability in long-COVID patients and healthy control. But now we have moved a bit forward, and we are trying to understand those who respond or not to ivabradine, because we have some heart rate variability domains that are typical of parasympathetic tone and other of sympathetic tone. Very briefly, I want to show you that, for example, those patients having an imbalance, there are some patients having an imbalance toward a sympathetic tone. Now we are understanding from our heart rate variability studies, and some other patients conversely in the heart rate variability sub-analysis have already a dysautonomia toward the parasympathetic tone. So this is probably, we have some patients that are responding to ivabradine, and some patients that are not responding to ivabradine. And the interesting thing was that those patients responders to ivabradine therapy had all of them an imbalance towards a sympathetic tone. So initially, when we treated, we gave ivabradine along the other non-pharmacological interventions to everyone. But now, most probably, we will try to personalize more the ivabradine approach. So this is literally the basic first line thing we do to all patients with long COVID. And if they have the criteria for, as Vita was saying, it's not only POTS, the rigorous POTS, but the spectrum of orthostatic intolerance. We, along with non-pharmacological interventions, we use ivabradine to almost all the patients, literally much more than half of the patients have it. We use a dosage that goes from 2.5 to 7.5 twice a day. And we use it in the morning as soon as they wake up, and then in the afternoon, because initially we were prescribing morning and evening, but children don't need it before going to bed, because when they go to bed already rest, the heart rate goes down. And I learned from an Australian doctor that it's better to give 8 hours after the first dose when they are still in the middle of activities in the afternoon. The second step usually comes simultaneously with the first step, and what we do to our patients, of course, okay, many patients have chronic headache, chronic pains, insomnia, so these neurological symptoms. Some may also be really associated to dysautonomia, but anyway, in this case, we do an MRI to exclude like cancers, for example, EEG, electromyography, and the pseudoscan is a direct sign, non-invasive sign of small fiber neuropathy. So these patients, when they had chronic headache or these chronic pains in our clinic before the pandemic, or when these symptoms are not associated to COVID, our neurologists were always treating these patients with gabapentin. Gabapentin is historically known as an anti-epileptic drug, but has several different effects. It's used for several neuropathies, also have effects on mood, and we know that these children obviously have an impact on mood because they were having a perfectly normal life, and then they are not doing anything else at 12, 14, 15 years of age. This medication is always used in this condition when it is not related to COVID. When it was about COVID, doctors keep saying to patients it's due to the pandemic, lockdown. While if you go to the neurologist, they don't tell it's related to COVID. In our culture, most of these patients were receiving gabapentin. So we use a lot in all children who has chronic pain, chronic headache, and chronic headache can have a significant impact on daily life, of course. And so our second step, but most of the time, as these are different mechanisms, as I said, is along with the neurologists and this test, of course, to care for sleep, because as Vida was saying, it's not all about drugs, so several interventions, and gabapentin is the very second classic medication we use in our center. Then, as I was saying, so the step up, we do the step up every some months to see how patients respond to these therapies. And as I said, even this morning, usually we start the step one, first of all, in only the very severe cases, so not those children that have some fatigue after 3 months, but usually they are patients that arrived in our attention after being 2 or even 3 years with those severe symptoms. So I'm really talking about the most severe spectrum. In case those children have no or very little improvement with these medications, and we are facing really the most severe cases, we consider the third step. So we've talked a lot about endotheliopathy. I'm showing here just another study. So thromboinflammation is, it's documented. I don't think there are similar diseases in the history of medicine that have been studied with such advancing technologies and clearly confirmed by several independent cohorts like low COVID. Honestly, I don't recall any single disease. Pneumonia, that we talk about pneumonia as a disease, we know, but we literally in 2024 still don't know how properly define COVID-acquired pneumonia in children. And again, low COVID is documented with much better technology. So at the beginning of the pandemic, now we've stopped for a number of reasons because we were not seeing this anymore with Omicron. We were using the lung perfusion studies, which were also used in adults, to have something to justify ourselves to use anticoagulation in case we found hypoperfusion in one or in some areas of the lung in these children, using this as kind of an indirect sign of endothelial dysfunction, and also thinking that if this was going on in the lung microcirculation, this might be happening also in other tissues on the body. And what we do something different in compared to what Vida told us, because we here again taper our approach. Our first choice after discussing with our coagulation specialist was to focus on enoxaparin or now the oral derivative rivaroxaban, because they not only are anticoagulants, but we have tens of studies showing that enoxaparin has clear anti-inflammatory and antioxidant effects even on the endothelial level. So our hypothesis was that the platelet activation in the microclots are probably triggered by chronic inflammation and endothelial inflammation. So to be cautious, we said, okay, try to target the endothelial level, which will in turn trigger the infection. So we start with enoxaparin at a prophylactic dose, again not at anticoagulation dose, meaning half the dose of the classic. We use enoxaparin in several other pediatric conditions. So we use it at the prophylactic dose because our approach is that we don't have a classic thromboembolic event. And now, since a while, we are slowly shifting to using rivaroxaban, who has is an oral formulation and has the same effects, even in terms of endothelial and anti-inflammatory effects, as with enoxaparin. just in a few cases where we didn't have any response to the enoxaparin, we also did aspirin, but we never arrived to triple medications. The other step, we do have, my feeling is that actually with Omicron and post-Omicron waves, we are having more neurocognitive symptoms, more similarly to adults, but this is only my personal experience. And in my opinion, the brain fog, the memory issues are those that are the most difficult symptoms to be treated in my experiences. So in case children do not respond to the first 3 steps, again, we said, let's see what the literature tells us. These are 3 studies that actually replicated each other, what David Cucino was telling us this morning. So basically 3 studies who took antibodies from long COVID patients with neurological, neuropsychiatric symptoms, putting them, in 2 studies to mice, and in another study with a muscle prototype, and in all 3 studies they replicated the adult symptoms to mice, and in the other model, in this model here, antibodies put on a muscle prototype generated inflammation in this muscle prototype. So it's clear there is something about antibodies or something in the sera of this patient. So again, what is the easiest thing to do for me as a pediatrician? There are, of course, biological agents that who clearly trigger the immune system and antibodies or whatever, but these are too difficult, of course, to and also have too many side effects to be used out of trials as off-label options. So in pediatrics, we have extensive use with IVIG. We use IVIG for almost everything. I use IVIG even in a 3-kilogram newborns with enterovirus infection, for example. In my opinion, they are very, very safe. They can have some side effects, but literally we use from Kawasaki disease, idiopathic inflammatory syndrome, multisystem inflammatory syndrome. So again, in the most severe cases, not responding to the first 3 steps, so meaning we start after 2 years of symptoms, we try everything for 1 year, nothing yet, we try monthly infusion. There are some little trials as well in the CFS, so David told us some patients respond, some not, but I'm showing the general step. And of course, if this also doesn't work, the next step is only a trial, we stop and we don't move forward. We talked a lot about T cells. This is one of my favorite studies of the long COVID pandemic. T cell infiltration has been demonstrated in several body tissues and sanctuaries. So this is again not a theory, something proven. And I'm really as well optimistic that some biological agents who can target both antibodies, antibodies, and T cells can be really promising, but this will only be about trials. And of course, since the beginning, patients are instructed about pacing, already Vinita told us, and of course, patients are supported from a mental health perspective because their life is completely disrupted. But we also try to inform schools, but many times even the family to recognize the disease because it's not even obvious from parents, mostly from fathers to recognize the disease as real. This is also why I wrote the book, literally for parents and families and school teachers. So as even in our experience, most children actually do quite well. We have several, I'll show you something later, several good experiences. I don't know if compared to adults, it's because we start earlier or literally I don't know why, or because we use more medications, I don't know. But the question is how to step down, of course I don't know. So basically, if children improve, and improving again, it's very easy. They didn't go to school and they come back to go to school most of the time. They were not able to do 1 floor of stairs and they can go out or do some sport or go out with friends. So if they provide this sort of self-reported improvements, we actually also monitor with cardiopulmonary exercise testing and 24 hours electrocardiogram. and most of the time, I will show maybe some picture later. If they also these objective measures are improved, we reduce from the most difficult medications as going back, which is of course starting from the anticoagulation. We reduce the doses, then we remove. Again, if symptoms remain, if the patients remain feeling well, we recheck with under the lighter medical treatment, the objective measures, if okay, remove like that. So this is a process again that may take several months. This is just one of the examples. This is a girl who was a horse rider. During the lockdown, she was very happy actually because she owned horses, so she was not going to school and she was horse riding every day. Again, developed long COVID actually when schools reopened, and she was said, okay, it's psychological because of the lockdowns, but she said, no, I was super happy going riding my horses. Anyway, eventually we treated, as you saw, the cardiopulmonary exercise testing. She was not able to finish the effort, had indirect parameters of pulmonary hypertension, and then with the treatment, she was all the parameters improved, and eventually she was even able to tolerate to conclude the test with a stronger protocol, as you can see. Or for example, this is the same girl with this psychological condition, a simple 24-hour electrocardiogram. As you can see, during the day she was at school, so just walking or going to the toilet at school, she was almost always above 100 beats per minute. So we simply treated with non-pharmacological interventions with ivabradine. So here you can see the cutoff was 175, so the highest cutoff was 150. The heart rate is much more regular on a lower basic level. Okay, it's better, so we reduce the dose of ivabradine, and this is how we go on. Of course, there are many colleagues working with us doing this all. The only problem is each of us is doing anything else for 95% of the time, so it's our extra time. We do the long COVID patients because we are, for example, as pediatricians, and so we have to go to the emergency department several times a month. And there are several patients, of course, that are gaining a new life. This was the horse rider who started having a new life. This is my first patient from France who was able to do literally nothing and now was going out with friends. But I want to conclude because it was kind of a surprise. I received this letter just this morning from a patient again from France. The mother actually brought this to me this morning. I didn't expect. So, hi, Dr. Bonsenso. I swear I tell the patients to call me Danilo, but she wrote Dr. Bonsenso. I'm feeling better since the treatment. I know you have helped many children like me, and I want to thank you. I can do more things now than the last 3 years, and I feel less tired. So 3 years when we started everything. Recently, I went to Disneyland with my family. It would have been impossible a few months ago. Thanks for what you do. So, and that's it.